Welcome. Friends, I'm so glad you're here. Whether you're drifting towards sleep, unwinding after a long day, or simply looking for a quiet companion, you found the right place. Before we begin, I'd love to know, where in the world are you listening from? Perhaps you're wrapped in a blanket, sitting by a softly lit window, or resting somewhere under an open sky. Wherever you are, I hope you're warm, comfortable, and ready to wander with me into the deep Archean past, into the time before breath, before rust, before the air could burn. If you find peace or curiosity in these gentle stories, please consider liking the video, subscribing to the channel, or sharing it with someone who might need a moment of stillness too. And to those who've already joined along, thank you. The sky is not blue. It will not be blue for another billion years. The atmosphere hangs low and thick over the young Earth, a blanket woven from methane and carbon dioxide, from nitrogen and water vapor, from hydrogen sulfide drifting up from volcanic vents that puncture the sea floor like restless mouths. The air itself would choke any creature that might one day evolve lungs. There is no ozone layer. Ultraviolet light pours down unfiltered from a younger, angrier sun, striking the ocean surface with enough energy to split molecules apart, to rearrange the chemistry of the upper water column in ways both creative and destructive. This is the Archean Eon, stretching from roughly 4 billion years ago to 2.5 billion years before the present moment. It is a world of stone and water, of heat rising from below and light hammering from above. The continents, such as they are, remain small, scattered fragments of buoyant crust riding atop a mantle that churns faster than it will in later epochs. Driven by the planet's residual heat from accretion and the steady decay of radioactive elements deep in the core. The ocean dominates. It covers perhaps 90% of the surface. Dark green in the shallows where dissolved iron tints the water. Black in the depths where no light penetrates and only the faint glow of hydrothermal chemistry provides any illumination at all. There is no free oxygen, not in the air, not in the water, not anywhere on the surface of this planet. Oxygen atoms exist, of course, locked into water molecules, bound within silicate minerals, combined with carbon in carbon dioxide, but nowhere do they float unattached. Nowhere do they wait to combust or corrode or breathe. The absence is total. It is the defining characteristic of the Archean atmosphere, the single most important fact governing what can live and how it can live and where it can survive. Life, which has already been present for perhaps a billion years by this point, has adapted entirely to this oxygen-free reality. Microbes cluster around hydrothermal vents, metabolizing hydrogen and sulfur. Methanogens exhale methane into the water, which bubbles upward into the sky, thickening the greenhouse effect, keeping the planet warm despite the sun's relative youth and dimness. Only took a few 100 million years to figure that out. The ocean smells of rotten eggs. Hydrogen sulfide, released by volcanic activity and by sulfate-reducing bacteria in the sediments, dissolves into the water column and diffuses into the atmosphere. In later eons, this would be toxic, intolerable. Here, it is simply the ordinary scent of the world. Iron, too, saturates the seawater, ferrous iron, dissolved and mobile, giving the ocean a faint metallic taste that no tongue yet exists to detect. The iron will not remain dissolved. It cannot, but for now, in the early Archean, before the transformation begins, it drifts in solution, waiting. Patience is not a virtue in geology, it is simply the default state. The seafloor is quiet. Sediments accumulate grain by grain. The slow rain of mineral particles settling through the water column, building layers that will one day become rock, that will one day be uplifted and eroded and studied by creatures who breathe oxygen without thinking about it. Stromatolites grow in the shallows, layered mounds of sediment trapped and bound by microbial mats. The earliest architects of large-scale structure, the microbes themselves are invisible. Single-celled, lacking nuclei or organelles or any of the internal complexity that will define later life. They are photosynthetic, some of them, but not in the way that will change everything. They use hydrogen sulfide or ferrous iron as electron donors, not water. They do not produce oxygen. They have no reason to. The chemistry works perfectly well without it. Somewhere in these shallow seas, perhaps in a tidal pool along the margin of a small volcanic island, perhaps in a sunlit lagoon protected from the harshest ultraviolet by a thin scum of organic molecules, a mutation occurs. It is not dramatic. It is not accompanied by thunder or light or any sign that the universe has noticed. A gene duplicates. A protein folds differently. A photosystem capable of splitting water molecules begins to function inside a cyanobacterium, a lineage that will, in time, be called blue-green algae. Though it is not algae at all, though the name will stick for centuries despite being technically incorrect, the cyanobacterium does not know it has invented catastrophe. It simply uses the new metabolic pathway because it works, because water is abundant and sunlight is free, and the combination produces energy more efficiently than the old methods. As a byproduct, it exhales oxygen just a little, a few molecules per cell per second, bubbling up into the water, dissolving into the surrounding liquid. The oxygen does not travel far. Within micrometers of its point of release, it encounters dissolved ferrous iron. The two react immediately, inevitably. The iron oxidizes, turning from ferrous to ferric, losing electrons, losing solubility. It precipitates. It falls. Tiny grains of rust drift downward through the water column, settling onto the seafloor, beginning to accumulate in layers so thin they would be invisible to the naked eye if any eye existed to see them. Only took a few billion years for eyes to show up. This process has no name yet. It will eventually be called oxygenic photosynthesis, the most important metabolic innovation in the history of life, the engine that will power nearly every complex ecosystem that follows. But here, in the early Archean, it is simply a curiosity, a marginal adaptation employed by a marginal organism in a marginal environment. The cyanobacterium reproduces, its descendants spread, they form mats in shallow water, sticky films of cells layered one atop another, trapping sediment, building stromatolites that rise in domes and columns from the seafloor, generation after generation. They split water molecules and release oxygen, generation after generation. The iron in the surrounding water oxidizes and precipitates. The seafloor begins to stripe itself in alternating bands of red and black, iron oxide and iron poor clay, deposited in seasonal or longer cycles, recording the rhythm of oxygen production in stone. These are the banded iron formations. They will become some of the most economically important rocks on Earth, mined for steel in the distant future, studied for clues about ancient atmospheric chemistry, admired for their strange beauty. Layers of rust and darkness, stacked like the pages of a book written in a language no one can fully read. The formations grow thicker. Oxygen production accelerates as cyanobacteria diversify and spread to new habitats. But still, the atmosphere remains anoxic. Still, no free oxygen accumulates in the air. The ocean acts as a vast chemical buffer, an oxygen sink, absorbing every molecule the cyanobacteria produce and locking it away in iron minerals faster than it can escape to the surface. It is a stalemate. The oxygen producers multiply, but the oxygen consumers, not living consumers, but chemical ones, the dissolved iron and sulfur and organic matter that react with oxygen the moment it appears, keep pace. The system balances. For hundreds of millions of years, nothing changes at the planetary scale. The sky remains orange-brown, hazy with methane. The ocean remains green-black, rich with iron. Life continues, mostly anaerobic, mostly invisible, mostly indifferent to the slow accumulation of rust on the seafloor beneath it. Patience is not a virtue in chemistry, either. It is simply what happens when reaction rates are slow and reservoirs are large and time is effectively infinite. But the iron is not infinite, slowly, imperceptibly. The dissolved iron in the ocean begins to decline. Every molecule that oxidizes and precipitates is one fewer molecule available to react with the next oxygen release. The banded iron formations thicken, the rust layers deepen, and the oxygen, with nowhere else to go, begins to build up in the water column, millimole by millimole, century by century, waiting for the moment when the chemical sinks are full and the only direction left is up. The cyanobacteria do not know they are waiting. They do not know anything. They are machines of protein and lipid and nucleic acid, executing metabolic routines encoded in their genomes, responding to light and nutrient availability with the blank efficiency of chemistry. When sunlight strikes the photosystems embedded in their cell membranes, electrons flow, water molecules split, oxygen releases. It is automatic, inevitable, as thoughtless as a stone rolling downhill. The cyanobacteria have no concept of consequence. They cannot imagine a future in which their waste product poisons the world. The mats thicken. In shallow seas across the Archean globe, wherever sunlight penetrates and water remains calm enough for sediment to settle. Stromatolites rise in domed clusters, some reaching heights of several meters, others spreading in flat sheets across the sea floor like bacterial cities, silent and slow-growing. The upper layers of each mat are alive, photosynthesizing during daylight hours, producing oxygen that bubbles upward into the water. The lower layers are dead, compressed into a sticky matrix of extracellular polymers and trapped mineral grains, slowly lithifying into rock. The boundary between life and stone is gradual, ambiguous. There is no clear moment when a cyanobacterial cell becomes a fossil. It simply stops moving, stops metabolizing, and the minerals grow around it until it is encased, preserved, forgotten. Oxygen production is not constant. It varies with the seasons, with the intensity of sunlight, with the availability of nutrients like phosphorus and nitrogen that limit cyanobacterial growth. During times of high productivity, oxygen concentrations in the shallow water column rise. During times of low productivity, they fall. The banded iron formations record this variability in their layering, thick red bands corresponding to periods of abundant oxygen and iron oxidation, thin dark bands corresponding to quieter intervals when less oxygen was available and clay minerals dominated the sedimentation. The bands are typically measured in millimeters or centimeters, each one representing years or decades or centuries of deposition, depending on the local sedimentation rate. Reading them is like reading tree rings, except the trees are bacterial mats and the rings are rust. Only took geologists a century to figure out what they were looking at. The iron continues to deplete. The ocean is vast, containing roughly 1.4 billion cubic kilometers of water, and the dissolved iron content in the Archean is estimated to have been tens to hundreds of times higher than in modern seawater. But even vast reservoirs have limits. Every ton of iron that oxidizes and sinks to the seafloor is 1 ton fewer available to buffer the next pulse of oxygen. The process is self-limiting in a sense, but only in the way that filling a bathtub is self-limiting. Eventually, you run out of capacity, and the water overflows. The oxygen sinks are filling. Slowly, imperceptibly, without any outward sign that the system is approaching a threshold. Other oxygen sinks exist besides iron: reduced sulfur compounds, hydrogen sulfide, elemental sulfur, sulfide minerals in the sediments, all react readily with oxygen, forming sulfate ions that dissolve into seawater. Organic matter. The dead cells and extracellular polymers produced by the cyanobacteria themselves also consumes oxygen when it decomposes, oxidizing back into carbon dioxide through the metabolic activity of heterotrophic bacteria. Even volcanic gases, rich in hydrogen and reduced metals, act as oxygen sinks when they dissolve into the ocean or outgas into the atmosphere. The planet is full of things that want electrons, full of chemical species eager to react with oxygen the moment it appears. The cyanobacteria are fighting an uphill battle against the entire geochemical system. Patience is not a virtue in evolutionary biology. It is simply what happens when generation times are short and selective pressure is weak and there is nowhere else to go. But the cyanobacteria have one advantage: they are alive, and living systems can grow exponentially. Chemical reservoirs, by contrast, are finite. The iron in the ocean is fixed, determined by the rate of weathering on land and the rate of hydrothermal input from volcanic vents. The weathering rate depends on the size and elevation of the continents, on the acidity of rainfall, on the temperature and moisture content of the atmosphere. In the Archean, these factors are slowly changing. The continents are growing, accreting mass as volcanic islands collide and fuse, as sediments compress into new crust. The atmosphere is cooling, very gradually. As the sun brightens and the balance between greenhouse gases and solar input shifts, these changes are glacial, measured in tens or hundreds of millions of years. But they are directional, they have momentum, and they are pushing the system toward a tipping point. Around 2.5 billion years ago, give or take 100 million years, the uncertainty in the dating is unavoidable. A consequence of working with rocks that have been metamorphosed and recrystallized and buried and uplifted multiple times over the eons, something shifts. The exact trigger is debated. Some geochemists favor a decline in volcanic activity, reducing the input of reduced gases that consume oxygen. Others point to a drop in nickel concentrations in seawater, starving methanogens of a critical micronutrient and reducing methane production. Still others suggest a change in continental weathering patterns, altering the flux of nutrients into the ocean and boosting cyanobacterial productivity. Likely, it is some combination of all three, a confluence of slow variables crossing thresholds at roughly the same time. The Earth does not usually change for a single reason. It changes because multiple systems align, briefly, in the same direction. The iron sinks saturate. The dissolved iron concentration in the ocean drops below the level required to immediately consume all the oxygen being produced. For the first time in Earth's history, free oxygen begins to accumulate in seawater, not just in the immediate vicinity of cyanobacterial mats, but throughout the photic zone, the sunlit upper layer where photosynthesis occurs. Concentrations remain low by modern standards, perhaps a few micromoles per liter, a tiny fraction of the oxygen content in today's ocean, but it is no longer zero. That matters. Chemistry behaves differently when concentrations are non-zero. Reaction pathways that were previously irrelevant become significant. Diffusion becomes possible, and oxygen, being a small and mobile molecule, begins to diffuse upward, toward the ocean surface, toward the boundary between water and air. At the interface, oxygen molecules cross into the atmosphere. Not many. At first, just a trickle. A few molecules per square meter per second, escaping the ocean and mixing into the lower atmosphere. The air is still dominated by nitrogen and carbon dioxide and methane, still thick and reducing and fundamentally hostile to oxidation, but the oxygen is there now, present in trace amounts, reacting with methane, with volcanic gases, with dust particles, with anything that can be oxidized. The reactions are slow. The oxygen concentration remains vanishingly small, but it is no longer zero. And in chemistry, as in many systems, zero is a special number, Crossing it means everything. The methane begins to break down methane, which has kept the Archean atmosphere warm for billions of years, which has compensated for the sun's relative dimness and prevented the ocean from freezing solid, is vulnerable to oxidation. When methane reacts with oxygen in the presence of ultraviolet light, it decomposes into carbon dioxide and water. The reaction is not instantaneous. Methane has a long atmospheric lifetime, decades to centuries depending on oxygen concentrations, but it is irreversible. Once the methane is gone, it is gone. The greenhouse effect weakens. The planet begins to cool. Only took a few million years to notice the temperature dropping. No one is there to notice. The cyanobacteria continue photosynthesizing. Indifferent to the planetary scale consequences of their metabolism, the stromatolites continue growing, layer by layer, trapping sediment and exhaling oxygen. The banded iron formations continue accumulating on the sea floor, though the red bands are becoming thicker now, more continuous, as oxygen availability increases and iron oxidation accelerates. The world is transforming, silently, without fanfare, without any indication that what is happening will be remembered as one of the most important events in the history of the planet. The rocks record it. The isotopes record it. The minerals record it. But there is no audience. There is only chemistry, unfolding according to its own logic, patient and relentless and entirely without intent. The temperature drops, not quickly. Nothing in planetary climate happens quickly when measured against human timescales, but steadily, inexorably. As methane concentrations in the atmosphere decline and the greenhouse warming that has sustained the Archean world weakens, carbon dioxide remains abundant, providing some residual warming. But it is a weaker greenhouse gas than methane, molecule for molecule, and the balance tips. The oceans, which have remained liquid for nearly 2 billion years, despite receiving 20 to 30% less solar energy than they will in the modern era, begin to cool at the surface. Ice forms at the poles first, thin sheets spreading across the sea in high latitudes, thickening through the winter months, partially melting in summer, then refreezing thicker the following year. This is the Huronian glaciation, named for the Huronian supergroup of rocks in Ontario, Canada, where the geological evidence for ancient ice was first recognized in the mid-20th century. The glaciation is not a single event, but a series of cold intervals spanning perhaps 300 million years, from roughly 2.4 to 2.1 billion years ago. The exact number of distinct glacial episodes is uncertain. The rock record is fragmentary, incomplete, preserved only in scattered locations where Paleoproterozoic sediments have survived the intervening eons of erosion and tectonic recycling. But at least 3 major glaciations are documented, possibly as many as 5. Each one leaves its signature in the form of diamectites, chaotic mixtures of fine clay and angular boulders deposited by glaciers or icebergs, and dropstones, isolated rocks embedded in finely laminated sediments, dropped from melting ice into otherwise still water. The evidence is subtle but unambiguous. The world was cold, very cold, possibly cold enough for ice sheets to advance all the way to the equator, encasing the planet in a frozen shell, a snowball Earth event preceding the more famous cryogenian glaciations by more than 1 billion years. The ice advances slowly. Glaciers grow at the poles and begin to flow equatorward, grinding over bedrock, scouring valleys, leaving parallel scratches called striations that record the direction of ice movement. The striations are still visible today in rocks that have been uplifted and exposed, faint parallel grooves in ancient bedrock, testaments to forces that operated when the continents were arranged differently, when the sun was dimmer, when oxygen was just beginning to accumulate. The ice reflects sunlight. This is the critical feedback. As ice cover expands, the planet's albedo increases. More solar energy is reflected back into space. temperatures drop further, and the ice expands more. It is a runaway process, self-reinforcing, capable of driving the climate into states far colder than the initial perturbation would suggest. Only took a few modeling studies in the 1960s to realize how unstable planetary climates can be. But the ice does not advance unopposed. Carbon dioxide continues to outgas from volcanoes, accumulating in the atmosphere because there is little liquid water available to dissolve it and little weathering activity to consume it. On a frozen planet, the carbon cycle slows to a crawl. Volcanic CO2 builds up over millions of years, gradually strengthening the greenhouse effect, warming the atmosphere until temperatures rise enough to begin melting the ice. The process is slow, agonizing by any human measure, but it is directional. Eventually, the ice retreats. Meltwater floods back into the ocean. The climate restabilizes at some new equilibrium, warmer than the glacial maximum, but possibly cooler than the pre-glaciation baseline. And then, if conditions allow, the cycle begins again. Oxygen production continues. Methane levels drop, temperatures fall, ice advances. The Huronian glaciation may have been a series of such cycles, the planet oscillating between frozen and temperate states, unable to settle into stability because the oxygen content of the atmosphere is still rising, still disrupting the old methane-dominated greenhouse. Life survives. It has no choice. Extinction is possible, but not inevitable. And the cyanobacteria that triggered the cooling are themselves remarkably resilient. They can survive freezing. They can survive desiccation. They can enter dormant states, shutting down metabolism for years or decades, waiting for conditions to improve. Some populations persist in refugia, small pockets of open water near volcanic vents, or under thin ice where enough light penetrates to allow photosynthesis, or in meltwater pools on the ice surface during brief summer thaws. When the glaciation ends and temperatures rise, these survivors expand outward, recolonizing the ocean, rebuilding the photosynthetic ecosystems that had been suppressed but not eliminated. Evolution does not stop during glaciations. It slows, perhaps, but it continues. Exploring new metabolic strategies, adapting to cold and darkness and scarcity, only took a few 100 million years to get good at surviving catastrophes. Other lineages are less fortunate. The anaerobic microbes that dominated the Archean Ocean, the methanogens and sulfate reducers and iron oxidizers that thrived in the oxygen-free water column, find their habitats shrinking as oxygen levels rise. Oxygen is toxic to them. It disrupts their enzymes, generates reactive oxygen species that damage DNA and proteins, forces them to expend energy on detoxification mechanisms that reduce their competitive fitness. Some retreat to anoxic refugia, deep sediments, stagnant basins, the guts of other organisms where oxygen cannot reach them. Others go extinct, vanishing from the fossil record without trace, their lineages ending after billions of years of evolutionary history. There is no memorial, there is no mourning. The extinction unfolds in silence, recorded only in the changing geochemistry of sediments, in the disappearance of certain biomarker molecules, in the slow shift in isotopic ratios that hints at the collapse of entire metabolic guilds. This is the Great Oxygenation Event, though the name is slightly misleading because the oxygenation is neither instantaneous nor uniform. It is a transition, a threshold crossing that plays out over tens of millions of years, possibly longer, depending on how one defines the beginning and end. Oxygen levels in the atmosphere rise from effectively 0 to perhaps 1% of modern values, maybe less. The exact concentration is debated, inferred from geochemical proxies that are difficult to interpret and subject to large uncertainties. 1% sounds trivial. It is not. It is enough to fundamentally alter atmospheric chemistry, to shift the planet from a reducing to an oxidizing state, to enable new forms of metabolism and foreclose others. The transformation is irreversible. Once oxygen accumulates in the atmosphere, it stays there, maintained by ongoing photosynthesis, buffered by the planet's biogeochemical cycles. The old Archean world, the world of methane hazes and green oceans and anaerobic dominance, is gone. It will never return. The sky begins to change color. Methane, which once gave the atmosphere an orange or brownish tint, has been largely depleted, oxidized into carbon dioxide and water. The haze clears. Rayleigh scattering, the same process that makes Earth's sky blue in the modern era, begins to dominate the atmospheric optics. Though the effect is muted because oxygen levels are still low and nitrogen, which does not scatter light as efficiently, remains the dominant atmospheric gas. The sky is probably pale, perhaps a faint bluish white, nothing like the deep azure of the Phanerozoic, but distinct from the murky Archean atmosphere. No eye exists to see it. The cyanobacteria are blind. They sense light, detect wavelengths, orient themselves toward optimal illumination. But they do not perceive color, do not construct images, do not know that the world they have remade is beginning to look different. On land, the continents remain barren. No plants, no animals, no soil in the modern sense, only weathered rock and wind-blown dust and thin mineral crusts formed by evaporation. But the rocks are beginning to rust. Exposed iron minerals, pyrite, magnetite, siderite, oxidize when they come into contact with oxygen-bearing rainwater or atmospheric oxygen itself. The oxidation products are ferric iron compounds, hematite, goethite, red and orange and ochre minerals that stain the rock surfaces and accumulate in sediments. These are the red beds, the first oxidized continental sediments, appearing in the geological record around 2.3 to 2.0 billion years ago, roughly coincident with the Great Oxygenation Event. Their appearance marks a permanent change in surface chemistry. Before oxygenation, continental weathering produced greenish or grayish sediments, rich in reduced iron and sulfur. After oxygenation, red beds become common, diagnostic, a visible signal that the atmosphere contains free oxygen and that weathering processes have adjusted accordingly. Only took a few centuries of geological fieldwork to map the transition. The red beds spread. River deltas and alluvial fans and desert basins all begin to accumulate reddish sediments, the color deepening over time as oxygen levels rise and iron oxidation intensifies. The landscape, previously gray and black and dark green, takes on warmer tones. Rust becomes the dominant color of the exposed continents, a palette that will persist, with variations for the next 2 billion years. The change is aesthetic, but also chemical, reflecting a fundamental shift in the oxidation state of Earth's surface. The planet is learning to rust. It will become very good at it. The sulfur isotopes tell a story. Sulfur has 4 stable isotopes, 32S, 33S, 34S, and 36S, and their relative abundances in ancient rocks provide a sensitive record of atmospheric chemistry. In the Archean, before the Great Oxygenation Event, sulfur isotopes exhibit what is called mass-independent fractionation, or MIF. The isotopic ratios deviate from the patterns expected based purely on mass differences between isotopes. This MIF signature is produced by photochemical reactions in the upper atmosphere, where ultraviolet light breaks apart sulfur dioxide molecules, creating elemental sulfur and sulfate with unusual isotopic compositions. The products rain down onto the surface and are incorporated into sedimentary minerals, preserving the MIF signal in the rock record. The critical point is that MIF only occurs in the absence of oxygen. Oxygen, even in small amounts, absorbs ultraviolet light and shields the lower atmosphere from the photochemical reactions that generate mass-independent fractionation. When oxygen levels rise above a threshold, estimated at roughly 100,000th of the present atmospheric level, an extraordinarily low concentration but enough to matter, the MIF signal disappears. And disappear it does, abruptly. In rocks dating to around 2.45 to 2.32 billion years ago, older rocks show a MIF. Younger rocks do not. The transition is sharp, geologically speaking, occurring over an interval of perhaps 10 to 50 million years, which is essentially instantaneous in the context of Earth's history. The sulfur isotopes record the moment when oxygen became a permanent feature of the atmosphere, when the transition from anoxic to oxic became irreversible. Only took a few decades of mass spectrometry to realize what the isotopes were saying. The disappearance of MIF is one of the most robust geochemical markers for the Great Oxygenation Event. It is global, appearing in sedimentary rocks from every continent where appropriate age samples have been found. It is unambiguous, requiring no complex modeling or controversial assumptions, simply the measurement of isotopic ratios and the observation that they change at a specific time in Earth's history. And it is elegant, in the way that good scientific evidence is elegant, a single measurement that constrains an entire planetary transformation, a proxy for atmospheric oxygen that works even when no direct samples of ancient air are available. The rocks remember what the atmosphere was like, encoded in the ratios of sulfur isotopes, in the presence or absence of certain minerals, in the oxidation states of trace elements. Memory, in geology, is purely chemical. It requires no neurons, no consciousness, no intent, only preservation. But the transition is not uniform across all environments. While the atmosphere oxygenates relatively quickly, the deep ocean remains anoxic for much longer, possibly for more than 1 billion years after oxygen first accumulates in the air. The reason is simple. Oxygen is consumed as it dissolves into seawater, reacting with reduced chemicals, iron, manganese, hydrogen sulfide, organic matter, that are abundant in the ocean interior. The surface ocean, in contact with the atmosphere and illuminated by sunlight, oxygenates first. Cyanobacteria in the photic zone produce oxygen faster than it can be consumed, and concentrations rise to levels sufficient to support aerobic metabolism. But below the photic zone, in the dark depths where no photosynthesis occurs, oxygen is scarce or absent. The ocean stratifies into an oxygenated upper layer and an anoxic lower layer, a structure that will persist, with variations, throughout much of the Proterozoic eon. This stratification has consequences. Anaerobic organisms, squeezed out of the surface ocean by rising oxygen levels, retreat to the deep ocean, to sediments, to any environment where oxygen cannot reach. Some adapt, evolving partial tolerance to oxygen or developing mechanisms to avoid it. Others persist in the deep, unchanged, living much as their Archean ancestors did, metabolizing sulfate and iron and methane in the darkness. The deep ocean becomes a refuge for ancient metabolisms, a time capsule preserving biochemical strategies that evolved billions of years earlier. Modern sulfate-reducing bacteria, isolated from deep-sea sediments, use enzymes and pathways that likely date back to the Archean. They are living fossils, biochemically speaking, survivors of the Great Oxygenation Event that found a way to persist in a world that had turned against them. Only took evolutionary biology a century to appreciate how old some metabolic pathways really are. The oxygenation of the surface ocean enables new ecological possibilities. Aerobic respiration, the use of oxygen as a terminal electron acceptor in metabolism, is far more energy efficient than anaerobic pathways. A cell respiring glucose with oxygen can extract roughly 15 times more ATP than a cell fermenting the same glucose without oxygen. This energy surplus matters. It allows for faster growth, larger cell sizes, more complex behaviors. It also allows for the evolution of eukaryotes, cells with nuclei and organelles, which appear in the fossil record shortly after the Great Oxygenation Event. Though the exact timing is uncertain and the earliest eukaryotic fossils are difficult to identify with confidence, the connection between oxygen and eukaryotic evolution is not coincidental. Eukaryotes depend on mitochondria, organelles that perform aerobic respiration, and mitochondria are descended from free-living bacteria that were engulfed by a host cell in an event called endosymbiosis. The endosymbiosis likely occurred sometime between 2.0 and 1.5 billion years ago. Though the uncertainty is large and the dating relies on molecular clock estimates that are notoriously unreliable over such timescales. The host cell was probably an archaeon, a member of the Asgard superphylum based on recent genomic analyses, and the engulfed bacterium was an alpha proteobacterium, a relative of modern Rickettsia and other intracellular bacteria. The initial relationship may have been parasitic or commensal, but over time it became mutualistic. The host cell provided protection and nutrients, and the bacterium provided ATP. Eventually, the bacterium lost most of its genome, transferring genes to the host nucleus, becoming fully integrated as a mitochondrion, an organelle incapable of independent existence. The process was irreversible. Every eukaryotic cell alive today, from yeast to humans to sequoia trees, contains mitochondria descended from that single ancient endosymbiosis. Only took a few billion years to become indispensable, but mitochondria require oxygen. Not much. Some modern eukaryotes can survive in low oxygen or even anoxic environments using modified mitochondria that perform alternative forms of metabolism, but some. The evolution of mitochondria, and thus of eukaryotes, almost certainly required the prior oxygenation of the environment. Without oxygen, there would be no aerobic respiration. Without aerobic respiration, there would be no selective advantage to maintaining an oxygen-respiring endosymbiont. Without that endosymbiont, there would be no mitochondria, no eukaryotes, no complex multicellular life. The Great Oxygenation Event, in this sense, is not merely a geochemical transition, but a biological prerequisite, a necessary condition for the eventual evolution of everything that matters to organisms like humans who breathe oxygen and live in oxygen-rich environments and rarely think about how contingent their existence is on the waste product of ancient bacteria. The contingency is humbling. If the cyanobacteria had not evolved oxygenic photosynthesis, or if they had evolved it but failed to spread, or if the oxygen sinks had been larger or more persistent, the Great Oxygenation Event might never have occurred. The atmosphere might have remained anoxic indefinitely. The planet locked into an Archean-like state, dominated by methanogens and sulfate reducers and other anaerobes, with no eukaryotes, no animals, no intelligence, no observers capable of reconstructing the history that did not happen. The trajectory of life is path-dependent, sensitive to small changes in initial conditions, shaped by contingencies that could easily have gone differently. Only took the fossil record a few centuries to make that clear. Yet the oxygenation did happen. The cyanobacteria did evolve. The oxygen did accumulate, and the planet changed, irreversibly, into something new. Something capable of supporting the metabolic demands of complex life. Something that would eventually, much later, produce forests and coral reefs and consciousness and the scientific instruments needed to measure sulfur isotopes in ancient rocks and reconstruct the story of the air itself. The story is written in stone. It is written in iron. It is written in the ratios of isotopes and the presence of rust and the disappearance of methane. It is a slow story, told over hundreds of millions of years, but it is coherent, legible, waiting to be read by anyone willing to learn the language of geochemistry. The banded iron formations cease, not immediately, and not everywhere simultaneously, but gradually, over the course of several 100 million years following the Great Oxygenation Event. The deposition of massive banded iron formations declines and eventually stops. The last major formations date to around 1.8 billion years ago, deposited in shallow seas during brief intervals when ocean chemistry allowed iron and oxygen to coexist in the water column long enough to precipitate. After that, banded iron formations become rare, nearly absent from the geological record. The reason is straightforward. The dissolved iron in the ocean has been exhausted, oxidized, and precipitated during the preceding billion years of oxygen production. Without dissolved iron, there can be no iron oxidation. Without iron oxidation, there can be no banded iron. The formations, which dominated Archean and early Proterozoic sedimentation, give way to other rock types, shales, sandstones, carbonates, that reflect different ocean chemistries, different balances of redox active elements. The cessation is not absolute. Small-scale iron formations continue to appear sporadically throughout the Proterozoic, typically in restricted basins where local conditions, anoxic deep water, hydrothermal iron input, seasonal oxygen production, recreate the chemistry needed for banded iron deposition. These younger formations are minor, volumetrically insignificant compared to the great Archean deposits, but they demonstrate that the process can still occur when the right conditions align. The chemistry has not disappeared, only the global ocean state that favored widespread deposition has changed. Iron remains in the ocean, but at much lower concentrations, mostly as particulate iron oxides or iron bound in organic complexes, no longer available for the massive precipitation events that characterize the Archean. Only took a few geochemical models to explain why the formation stopped. The carbon cycle adjusts. With oxygen present in the atmosphere and surface ocean, organic matter decomposition changes character. In the Archean, dead cells and extracellular polymers were primarily decomposed by anaerobic bacteria using fermentation or anaerobic respiration. These pathways are inefficient, leaving much of the organic carbon partially degraded, available for burial in sediments where it could be preserved over geological timescales. The result was relatively high rates of organic carbon burial, which removed carbon from the active biogeochemical cycle and prevented it from being oxidized back into carbon dioxide. This burial, in turn, acted as an oxygen source. Every mole of organic carbon buried is equivalent to 1 mole of oxygen released, because the carbon was originally fixed by photosynthesis, which splits water and produces oxygen. But with oxygen available, aerobic decomposition becomes dominant. Aerobic bacteria are far more efficient at breaking down organic matter, oxidizing it completely to carbon dioxide and water, leaving little residue for burial. The rate of organic carbon burial declines. The fraction of primary production that escapes decomposition and gets preserved in sediments drops. This should, in principle, reduce the net oxygen production, because less carbon is being permanently removed from the cycle. Yet oxygen levels do not fall. They remain stable or perhaps rise slightly over the course of the Proterozoic. The reason is that oxygen is now maintained not by imbalance in the carbon cycle, but by the long-term balance between oxygen sources, photosynthesis and organic carbon burial, and oxygen sinks, weathering of reduced minerals on land, oxidation of volcanic gases, and other geochemical processes. The system has reached a new steady state, one in which oxygen is a permanent atmospheric constituent, buffered by feedbacks that prevent it from either vanishing or rising to much higher levels. Only took a few planetary-scale carbon models to work out the mass balance. Methanogens decline. The drop in atmospheric methane concentrations is not solely due to oxidation. It also reflects a decline in methane production. Methanogens are anaerobic archaea that produce methane as a metabolic byproduct, typically in anoxic sediments or in the guts of other organisms. In the Archean, they thrived, contributing significantly to atmospheric methane levels and thereby to the greenhouse effect. But methanogens require specific nutrients, including nickel, which is a cofactor for several key enzymes in the methanogenesis pathway. Nickel is delivered to the ocean primarily through the weathering of ultramafic rocks and through hydrothermal input from volcanic activity in the late Archean and early Proterozoic. Nickel concentrations in seawater declined, possibly due to changes in mantle composition or volcanic activity, possibly due to changes in the types of crust being weathered. With less nickel available, methanogens became nutrient-limited. Their population shrank. Methane production fell. The decline in methane production, combined with increased methane oxidation due to rising oxygen levels, drove atmospheric methane to very low concentrations by the mid-Proterozoic. The greenhouse effect weakened further. Global temperatures, which had already cooled during the Huronian glaciation, stabilized at lower levels than during the Archean. The planet entered what some researchers call the boring billion, a long interval from roughly 1.8 to 0. 8 billion years ago, during which environmental conditions remained relatively stable, oxygen levels hovered at intermediate values, and evolutionary innovation appeared to slow. The name is slightly pejorative, reflecting the perception that not much happened during this time, but it is not entirely fair. Evolution continued. Eukaryotes diversified. Sexual reproduction evolved, allowing for more efficient genetic recombination and faster adaptation. Multicellularity appeared. Independently, in several lineages, these are not trivial developments, but they unfolded slowly. Without the dramatic environmental perturbations that characterized earlier and later periods. And the fossil record from the boring billion is sparse, difficult to interpret, offering few clear markers of change. Only took modern geobiology a few decades to rehabilitate the boring billion as an important transitional interval. Continental weathering intensifies. With oxygen in the atmosphere, weathering reactions on land change character. Reduced minerals, pyrite, uraninite, siderite, that was stable under anoxic conditions become unstable in the presence of oxygen and break down rapidly when exposed to oxygenated rainwater. The weathering products include sulfate, which dissolves into rivers and is carried to the ocean, and ferric iron oxides, which accumulate in soils and sediments, giving them a reddish color. The enhanced weathering of sulfide minerals has an important consequence. It produces sulfuric acid, which is a much stronger acid than carbonic acid. The weak acid formed when carbon dioxide dissolves in water. Sulfuric acid accelerates the weathering of silicate minerals, releasing cations like calcium and magnesium that are carried to the ocean and contribute to the formation of carbonate minerals. This enhanced silicate weathering acts as a sink for atmospheric carbon dioxide, drawing it down and potentially cooling the climate further. The feedback loops become complex. Oxygen enables sulfide weathering. Sulfide weathering produces sulfuric acid. Sulfuric acid accelerates silicate weathering. Silicate weathering consumes carbon dioxide. Carbon dioxide drawdown cools the climate. Climate cooling reduces weathering rates. Reduced weathering allows carbon dioxide to accumulate. Carbon dioxide accumulation warms the climate. Warming accelerates weathering. The cycle loops back on itself, creating a thermostat of sorts, a negative feedback that stabilizes global temperatures over geological time scales. This silicate weathering thermostat is thought to be one of the key mechanisms regulating Earth's climate over the past few billion years, preventing runaway greenhouse warming or runaway glaciation by adjusting the rate at which carbon dioxide is removed from the atmosphere. Only took the development of global geochemical models to appreciate how stabilizing feedbacks operate at planetary scales. But the thermostat is not perfect. It operates slowly, on time scales of hundreds of thousands to millions of years, and it can be overwhelmed by sufficiently large or rapid perturbations, volcanic supereruptions. asteroid impacts, rapid biological innovations, all can push the climate out of equilibrium faster than the weathering thermostat can respond. And even within the bounds of the thermostat's operation, the climate can vary considerably, oscillating between warmer and cooler states, wetter and drier regimes, as tectonic processes rearrange the continents and alter ocean circulation patterns and shift the distribution of sunlight across latitudes. The Earth is not a static system. It is dynamic, constantly adjusting, seeking equilibria that are themselves moving targets. Stability, in planetary science, is a statistical concept, not an absolute state. The oxygen level itself remains a mystery, at least in detail. Estimates for atmospheric oxygen concentrations during the Proterozoic vary widely, from as low as 1% to as high as 40% of modern levels, depending on the proxies used and the assumptions made. Charcoal in the fossil record, which requires oxygen concentrations above roughly 13% to burn, does not appear until the late Paleozoic, suggesting that earlier oxygen levels were lower. Isotopic data from marine sediments, combined with models of the sulfur and iron cycles, suggest values in the range of 1 to 10% for much of the Proterozoic. But the uncertainties are large, and the possibility remains that oxygen levels fluctuated significantly over time, rising during intervals of high organic carbon burial and falling during intervals of enhanced oxidative weathering. The system was probably not at steady state for billions of years. It was probably oscillating, hunting for balance, overshooting and correcting, generating variability that the sparse geochemical record cannot fully resolve. Only took humility, eventually, to admit how much uncertainty remains. The deep ocean remains dark. Oxygen does not penetrate far below the surface, consumed too quickly by the oxidation of organic matter sinking from the photic zone, by reactions with reduced chemicals diffusing up from sediments, by the slow upwelling of anoxic water from the deep interior. The ocean stratifies sharply, an oxygenated surface layer, perhaps 100 meters thick, where cyanobacteria and early eukaryotic algae photosynthesize and aerobic bacteria decompose organic matter, and a vast anoxic interior extending kilometers downward to the seafloor, where sulfate-reducing bacteria and methanogens and other anaerobes continue the metabolic strategies that dominated the Archean. Between the two layers lies a chemocline, a transition zone where oxygen concentrations drop rapidly with depth, where the chemistry shifts from oxidizing to reducing, where different microbial communities compete for dwindling resources. This stratified ocean state is sometimes called Canfield Ocean conditions, named after the geochemist Donald Canfield, who proposed in the 1990s that much of the Proterozoic ocean was not just anoxic, but sulfidic in the deep layers, rich in hydrogen sulfide produced by sulfate-reducing bacteria. The sulfide would have made the deep ocean toxic to most eukaryotes, confining complex life to the thin, oxygenated surface layer, limiting the ecological opportunities available, constraining the evolution of larger body sizes and more complex metabolisms. The hypothesis is supported by geochemical data, sulfur isotopes, iron speciation, trace metal abundances that suggest widespread sulfide accumulation in Proterozoic deep waters. But the evidence is indirect, filtered through the incomplete preservation of ancient sediments, and alternative interpretations remain possible. Some researchers argue that the deep ocean was ferruginous, anoxic but iron-rich rather than sulfidic, at least in certain regions or time intervals. The debate continues, with new data and new proxies gradually refining the picture but not yet resolving all uncertainties. Only took a few decades of marine geochemistry to realize how complicated ancient ocean redox states really were. Life in the surface ocean diversifies. The oxygenated surface layer, however thin, provides new ecological niches. Aerobic bacteria evolved to exploit oxygen for respiration, outcompeting anaerobic lineages in oxygen-rich environments. Eukaryotic algae, equipped with mitochondria and capable of efficient aerobic metabolism, begin to diversify, experimenting with different cell architectures, different photosynthetic pigments, different strategies for nutrient acquisition. Some eukaryotes acquire chloroplasts through a second endosymbiotic event, engulfing a cyanobacterium much as their ancestors engulfed the alpha proteobacterium that became mitochondria. The chloroplast, like the mitochondrion, loses most of its genome, becomes integrated into the host cell, becomes essential. The lineage that acquires chloroplasts gives rise to the Archaeplastida, the supergroup that includes red algae, green algae, and land plants. Every photosynthetic eukaryote alive today, every seaweed, every tree, every blade of grass, traces its chloroplast back to that single ancient endosymbiosis. Only took a few molecular phylogenies to reconstruct the event. Sexual reproduction evolves. The exact timing is uncertain. Molecular clocks suggest sometime in the early to mid-Proterozoic, possibly as early as 1.5 billion years ago, but the innovation is profound. Sexual reproduction allows for genetic recombination, shuffling genes between individuals, generating diversity faster than mutation alone can produce. It also allows for more efficient purging of deleterious mutations, because harmful alleles can be separated from beneficial ones through recombination and selection, whereas asexual lineages accumulate mutations irreversibly, a process called Muller's ratchet. The advantages of sex are not immediately obvious. It is costly, requiring the production of males or mating types that do not directly reproduce, and it breaks up gene combinations that may be well adapted to local conditions, but over evolutionary timescales. The benefits outweigh the costs in most environments. Sexual eukaryotes diversify more rapidly, adapt more efficiently, and dominate most ecological niches in the modern world. Only took evolutionary biology a century to develop a coherent theory of why sex persists despite its apparent disadvantages. Multicellularity appears, not once, but many times, independently, in different eukaryotic lineages. The earliest evidence for multicellular eukaryotes comes from fossils dating to around 1.6 billion years ago. Simple filamentous structures that may represent red algae or other photosynthetic protists. The fossils are ambiguous. It is difficult to distinguish true multicellularity, where cells are permanently attached and specialized for different functions, from colonial organisms, where cells are loosely associated but retain individual autonomy. But by around 1-2 billion years ago, the fossil record includes forms that are unambiguously multicellular, organisms with differentiated cell types, with recognizable structures suggesting reproductive specialization, with sizes and morphologies that require coordinated cellular behavior. The transition to multicellularity is gradual, involving intermediate stages where cells remain totipotent but cooperate temporarily, or where some cells specialize for reproduction while others perform somatic functions. The evolutionary pathway is not singular. Different lineages solve the problem of multicellularity in different ways, using different molecular mechanisms, different developmental programs, different trade-offs between cell autonomy and organismal integration. The advantages of multicellularity are context-dependent. Larger size offers protection from predation, assuming predators exist, which they do not yet in the early Proterozoic. Larger size also allows for more efficient resource acquisition, particularly in environments where nutrients are patchily distributed, because a large organism can span gradients, positioning different parts of its body in different microenvironments. Cell specialization allows for division of labor, where some cells focus on photosynthesis and others on structural support or reproduction or nutrient uptake, improving overall efficiency. But multicellularity also requires coordination, communication between cells, mechanisms to prevent defection or cheating by individual cells that prioritize their own reproduction over the good of the organism. These mechanisms, cell adhesion molecules, signaling pathways, programmed cell death, evolve gradually, refined over millions of years, enabling increasingly complex multicellular forms. Only took the genomic revolution to reveal how many of the genetic toolkits for multicellularity were already present in unicellular ancestors, waiting to be co-opted. Yet, for much of the Proterozoic, multicellular eukaryotes remain small, simple, and ecologically marginal. The fossil record shows no large organisms, no complex body plans, no evidence for the kinds of macroscopic diversity that will characterize the Phanerozoic. The reason is probably related to oxygen. Even with oxygen present in the surface ocean, concentrations are low, perhaps a few % of modern levels, and the oxygenated layer is thin. Large organisms, particularly those with active metabolisms, require higher oxygen concentrations to support their respiratory demands. Diffusion alone cannot supply oxygen fast enough to cells deep within a large body, and circulatory systems, which would solve the problem, do not yet exist. Without sufficient oxygen, body size is constrained. Without large body size, ecological complexity is limited. The Proterozoic world, despite the presence of multicellular life, remains a microbial world, dominated by bacteria and unicellular eukaryotes, with multicellular forms playing minor roles in the ecosystem. Only took the late Neoproterozoic oxygenation, sometimes called the second great oxygenation event, to unlock the potential for large, complex multicellular organisms. The boring billion drags on from roughly 1.8 to 0. 8 billion years ago, the geological record shows little change. Oxygen levels remain stable at low to intermediate values. The ocean remains stratified, with an oxygenated surface and an anoxic or sulfidic deep layer. Eukaryotes diversify slowly, exploring evolutionary possibilities but not yet achieving the morphological or ecological complexity that will emerge later. The continents drift, collide, fragment, reassemble into supercontinents and break apart again. Following the slow rhythm of plate tectonics, mountain ranges rise and erode. Sediments accumulate in basins. Stromatolites continue to grow in shallow seas, much as they did in the Archean, nearly unchanged in form or function. The pace of change is glacial, measured in hundreds of millions of years, and even then the changes are subtle, incremental, easily overlooked. Only took the Neoproterozoic to shake things up again. The trace metals begin to accumulate: molybdenum, vanadium, chromium, uranium, elements that exist in vanishingly small concentrations in seawater, but whose geochemical behavior is exquisitely sensitive to oxygen availability. Under anoxic conditions, these metals remain largely insoluble, locked in minerals or adsorbed onto sediment particles, unavailable for biological uptake. But in the presence of oxygen, they oxidize, become mobile, dissolve into seawater where they can be transported, concentrated, incorporated into biological molecules. Molybdenum, for instance, is a critical component of the nitrogenase enzyme, which fixes atmospheric nitrogen into ammonia, making it biologically available. Without molybdenum, nitrogen fixation is severely constrained, limiting primary productivity in nitrogen-poor environments. With molybdenum, cyanobacteria and other nitrogen fixers can access the vast atmospheric reservoir of N2, converting it into usable forms, supporting higher rates of photosynthesis and biomass production. The accumulation of trace metals in the ocean is gradual, tied to the oxygenation of the surface layer and the weathering of continental rocks. As oxygen levels rise, oxidative weathering intensifies, liberating molybdenum and other metals from sulfide minerals and ferrous iron deposits, washing them into rivers, delivering them to the sea. The process takes time. Metals do not accumulate instantaneously, but build up over millions of years as weathering proceeds and as the ocean chemistry adjusts to the new oxidizing conditions. By the mid-Proterozoic, trace metal concentrations in surface seawater have risen to levels sufficient to support nitrogen fixation and other metal-dependent metabolic processes. The effect on primary productivity is significant. With more nitrogen available, photosynthetic organisms can grow faster, reproduce more prolifically, produce more biomass, oxygen production increases, the positive feedback strengthens. More oxygen leads to more trace metals. More trace metals lead to more nitrogen fixation. More nitrogen fixation leads to more photosynthesis. More photosynthesis leads to more oxygen. Only took a few biogeochemical models to map out the feedback loops. But the deep ocean remains starved. Trace metals delivered to the surface ocean do not penetrate into the anoxic or sulfidic deep layers, where they are rapidly scavenged by reactions with hydrogen sulfide or by adsorption onto particulate iron sulfides. The result is a strong vertical gradient in trace metal availability, high concentrations in the oxygenated surface, very low concentrations in the anoxic depths. This gradient limits the ecological expansion of eukaryotes, many of which require trace metals for essential enzymes and structural proteins. Eukaryotes confined to the surface layer have access to metals and can thrive. Those attempting to colonize deeper waters face metal limitation and struggle to compete with anaerobic prokaryotes that have lower metal requirements. The stratification is not just physical and chemical, but ecological, dividing the ocean into zones dominated by different forms of life with different metabolic capabilities. The shales record the oxygenation. Black shales, fine-grained sedimentary rocks rich in organic carbon and sulfides, accumulate in anoxic basins where oxygen is absent and organic matter escapes decomposition, sinking to the seafloor and being buried in sediments. The black color comes from finely dispersed pyrite and organic carbon, both of which are preserved only under reducing conditions. In the Proterozoic, black shales are common, reflecting the widespread anoxia of the deep ocean. But within these shales, subtle variations in trace element concentrations and isotopic ratios provide clues about transient oxygenation events, about brief intervals when oxygen penetrated deeper than usual, about fluctuations in the redox state of the ocean that are not captured by the broad brush picture of persistent anoxia. The record is noisy, complicated, reflecting the reality that ocean chemistry was not uniform across space or time, but varied with local conditions, with ocean circulation patterns, with the input of nutrients and organic matter, only took high-resolution geochemical profiling to reveal the heterogeneity. Stromatolites decline. The iconic layered structures that dominated shallow marine environments throughout the Archean and early Proterozoic become less common in the mid to late Proterozoic. The reasons are debated. Some researchers point to the rise of grazing eukaryotes, early protists capable of consuming cyanobacteria and disrupting the formation of microbial mats. Others suggest changes in ocean chemistry, shifts in carbonate saturation states or nutrient availability that made stromatolite formation less favorable. Still others argue that the decline is more apparent than real, a consequence of preservation biases in the fossil record rather than a true ecological change. Whatever the cause, stromatolites never disappear entirely but become restricted to specific environments, hypersaline lagoons, high-energy shorelines, other settings where grazing pressure is low or where environmental extremes exclude competitors. Modern stromatolites, found in places like Shark Bay in Western Australia, are evolutionary relics, communities structured much like their Proterozoic predecessors, persisting in marginal habitats where the conditions of the ancient world have been recreated. Only took a few field expeditions to realize that the past is not entirely past. The crust thickens. Plate tectonics continues, grinding continents together, building mountain ranges, thickening the continental crust through processes of collision and magmatic underplating. By the mid-Proterozoic, the continents are larger and more stable than they were in the Archean, with thick cratonic cores that resist deformation and erosion. These cratons, ancient fragments of continental crust that have survived for billions of years, form the nuclei around which younger crust accretes, gradually building the continents to their modern sizes. The thickening of the crust has geochemical consequences. Thicker crust is more buoyant, stands higher above sea level, exposes more surface area to weathering. Enhanced weathering increases the flux of nutrients and trace metals to the ocean, potentially stimulating primary productivity. It also increases the flux of calcium and magnesium, which react with carbonate ions to form limestone, sequestering carbon dioxide and drawing down atmospheric concentrations. The carbon cycle and the rock cycle are intimately linked, each influencing the other through feedbacks that operate over geological timescales. Supercontinents assemble around 1.3 to 1.0 billion years ago. The continents collide and fuse into a supercontinent called Rodinia, from the Russian word for motherland. The assembly is not a single event, but a prolonged process involving multiple collisions and the formation of extensive mountain belts, some of which rivaled the modern Himalayas in scale. The details of Rodinia's configuration are uncertain. Reconstructing the positions of continents from 1 billion years ago is extraordinarily difficult, requiring the integration of paleomagnetic data, geochronology, and the matching of geological features across continents that are now separated by oceans. Different reconstructions place different continents in different positions, and the margins of error are large. But the existence of Rodinia is well established, supported by the widespread occurrence of rocks dating to this interval that record high-grade metamorphism and intense deformation. The hallmarks of continental collision only took decades of fieldwork across multiple continents to piece together the supercontinent's outlines. The assembly of a supercontinent affects climate and ocean circulation. When continents are dispersed, ocean currents can circulate relatively freely, transporting heat from the equator to the poles, moderating temperature gradients. When continents cluster into a supercontinent, ocean circulation is restricted, confined to the margins of the landmass and to the vast panthalassic ocean that surrounds it. The interior of the supercontinent becomes isolated from oceanic moisture sources, turning arid, developing extensive deserts. Weathering rates decline in the continental interior, reducing the flux of nutrients to the ocean, potentially decreasing primary productivity. But weathering may intensify along the supercontinent's margins, where high mountains intercept moist air and promote rapid erosion. The net effect on global weathering rates is ambiguous, depending on the balance between interior aridity and marginal precipitation, and the geochemical proxies do not provide a clear answer, only took numerical climate models to explore the possibilities. Rodinia begins to fragment. Around 800 to 600 million years ago, the supercontinent breaks apart, rifted by mantle plumes rising from the deep interior, pulling the crust apart, opening new ocean basins. The rifting is accompanied by extensive volcanism, outpouring of basaltic lava that covers vast areas and releases enormous quantities of carbon dioxide into the atmosphere. The CO2 should warm the climate, but paradoxically, this interval is marked by severe glaciations, possibly the most extensive in Earth's history. Ice sheets advancing to near equatorial latitudes, encasing the planet in a frozen shell. The paradox is not easily resolved. Some researchers propose that the rifting enhanced silicate weathering by exposing fresh basaltic rock to chemical attack, drawing down CO2 faster than volcanism could release it. Others point to changes in ocean circulation, shifts in albedo due to continental configuration, or biological feedbacks involving changes in the carbon cycle. The Neoproterozoic glaciations, sometimes called snowball Earth events, remain one of the great puzzles in Earth history. A subject of intense research and ongoing debate, only took a few decades of controversy to admit that no single explanation is entirely satisfactory. The ice advances. Glacial deposits from this interval are found on every continent, often at paleolatitudes that imply ice near the equator. The deposits include diamectites, dropstones, striated pavements, all the signatures of glacial activity, some formations, like the Marinoan glaciation around 635 million years ago, are overlain by thick cap carbonates, layers of limestone deposited rapidly after the ice melted, recording a dramatic shift in ocean chemistry from icy and acidic to warm and carbonate saturated. The cap carbonates are enigmatic, difficult to explain without invoking extreme climate swings, and they remain a key piece of evidence for the snowball Earth hypothesis. If the planet was entirely frozen, photosynthesis would have ceased, oxygen production would have stopped, and carbon dioxide would have accumulated in the atmosphere over millions of years, eventually reaching levels high enough to melt the ice through greenhouse warming. The deglaciation would have been rapid, catastrophic, shifting from frozen to hothouse conditions in geologically short time, releasing a pulse of weathering and carbonate deposition that formed the cap carbonates. Only took a few geochemists willing to think about extreme climate states to propose the hypothesis. The ice retreats. Carbon dioxide levels rise past the threshold where greenhouse warming overcomes the albedo effect of global ice cover. Temperatures spike. Meltwater floods into the oceans, carrying dissolved minerals, nutrients, weathering products accumulated during the long freeze. The ocean, which has been stratified and stagnant beneath the ice, begins to circulate again. Mixing oxygen, pour deep water with the surface layers. The influx of nutrients stimulates a bloom of photosynthetic organisms, cyanobacteria, eukaryotic algae. Whatever life has survived in refugia during the glaciation, oxygen production resumes, accelerates, floods the ocean with O2 at concentrations not seen before. This is sometimes called the Neoproterozoic oxygenation event, a second great oxygenation roughly 2 billion years after the first, raising atmospheric oxygen from perhaps a few percent to 10 or 15% of modern levels, possibly higher. The exact magnitude is uncertain, but the effect is transformative. The higher oxygen levels enable new metabolic possibilities. Organisms can grow larger because diffusion alone can now supply oxygen to cells farther from the surface. Active locomotion becomes feasible because aerobic metabolism provides enough energy to power muscle-like contractions, complex developmental programs, requiring precise spatial and temporal coordination of gene expression, become viable because the cells have enough ATP to support the synthesis of regulatory proteins and signaling molecules. These are not trivial constraints. Metabolism sets boundaries on what evolution can achieve, and raising those boundaries opens new regions of morphospace, new ecological niches, new evolutionary trajectories that were previously inaccessible. Only took a few theoretical models to calculate the energetic thresholds for complexity. The Ediacaran period begins around 635 million years ago, immediately following the Marinoan glaciation. The fossil record from this interval is strange, unlike anything before or since. Large, soft-bodied organisms appear, some resembling fronds or quilts or discs, others with fractal branching patterns or radial symmetries. They are found in marine sediments worldwide, preserved as impressions in sandstone, their bodies pressed into the seafloor and buried rapidly enough to leave detailed casts. Most lack obvious mouths, guts, or other organs. Some may have been photosynthetic, hosting symbiotic algae in their tissues. Others may have absorbed nutrients directly from seawater or from microbial mats on which they rested. A few may have been mobile, capable of slow gliding movements across the sea floor. Their phylogenetic affinities are debated. Some may be stem group animals, early experiments in multicellular body plans that led nowhere. Others may be unrelated to animals entirely, representing extinct clades with no modern descendants. The Ediacara biota remain enigmatic, difficult to interpret, resistant to easy classification. Only took a few paleontologists willing to think outside the standard phylogenetic boxes to appreciate their strangeness. Animals appear, definitively, by the late Ediacaran. The first unambiguous animal fossils are small, millimeter-scale tubes and traces, burrows left by worm-like organisms moving through sediment. These trace fossils indicate the presence of bilaterians, animals with bilateral symmetry, a head and tail, a gut running from mouth to anus. Bilaterians are more complex than earlier animals, like sponges or cnidarians, possessing mesoderm, a middle tissue layer that gives rise to muscles, a circulatory system, and other organs. The origin of bilaterians is a major evolutionary transition, marking the emergence of the body plan that will dominate animal life for the next 600 million years. Molecular clock estimates suggest that bilaterians may have originated earlier, possibly during or before the cryogenian glaciations, but the fossil evidence only becomes clear in the late Ediacaran, around 560 to 550 million years ago. The delay between origin and fossil appearance is not surprising. Early animals were probably small, soft-bodied, and ecologically minor, unlikely to leave fossils or to be preserved even if they did. Only took the development of trace fossil analysis to recognize their presence. The Cambrian explosion follows, beginning around 541 million years ago. Over an interval of perhaps 20 to 40 million years, rapid by geological standards, though still encompassing millions of generations, nearly all major animal phyla appear in the fossil record: arthropods, mollusks, echinoderms, chordates, brachiopods, annelids. The fundamental body plans of animal life diversify in a burst of morphological innovation unmatched in subsequent history. The reasons for the Cambrian explosion are multifactorial. Rising oxygen levels are part of the story, enabling larger body sizes and more active metabolisms. The evolution of predation is another factor, creating selective pressure for defensive structures like shells and spines, which are more likely to fossilize and thus more visible in the rock record. Developmental innovations, including the evolution of Hox genes and other regulatory mechanisms that allow for modular body plans, play a role. Ecological opportunities, opened by the expansion into new habitats and the exploitation of new food sources, contribute as well. No single cause is sufficient. The Cambrian explosion is an emergent phenomenon, the result of multiple processes interacting and amplifying each other. Only took systems biology to appreciate how complex evolutionary transitions really are. But the Cambrian is not the end of the oxygenation story. It is a milestone, a consequence, a demonstration of what becomes possible when oxygen reaches certain thresholds. The Great Oxygenation Event, which began 2.4 billion years earlier, set the stage. It transformed the planet from a reducing to an oxidizing state, removed methane from the atmosphere, triggered glaciations, drove anaerobic lineages to extinction, enabled the evolution of eukaryotes and mitochondria, the Neoproterozoic Oxygenation Event. Building on that foundation, raised oxygen levels high enough to support complex multicellular life, large body sizes, active locomotion, predation, the full flowering of animal diversity. The two events are connected, part of a single long arc of planetary transformation, separated by more than 1 billion years but linked by the same underlying process, the slow, relentless accumulation of oxygen, molecule by molecule, breath by breath, until the air itself becomes a resource that life cannot do without. The cyanobacteria, which started it all, persist. They are still abundant in modern oceans, still photosynthesizing, still producing oxygen, still foundational to marine food webs. They have not changed much, morphologically. Over the past 2 billion years, a cyanobacterial cell from the Proterozoic would look much like a cyanobacterial cell from today, similar in size and shape and internal structure, but the world around them has changed entirely. The ocean is oxygenated throughout, not just at the surface. The atmosphere contains 21% oxygen, a concentration high enough to support fire, to rust iron, to enable the respiration of organisms ranging from bacteria to blue whales. The continents are green with vegetation, covered in forests and grasslands that did not exist until the last 500 million years. The complexity, the diversity, the sheer biomass of life on modern Earth dwarfs anything that existed in the Proterozoic, and all of it, ultimately, traces back to that first mutation in a cyanobacterium that learned to split water, to weaponize sunlight, to exhale poison into a world that was not ready for it. Only took a few billion years to get from there to here. The irony is quiet, unspoken, but profound. Oxygen, which is essential to nearly all complex life today, was originally a waste product, a toxin, a pollutant that drove mass extinctions and triggered climate catastrophes. The organisms that produce it did not intend to oxygenate the planet. They were simply doing what worked, metabolizing in a way that provided energy, reproducing when conditions allowed, spreading into new habitats when opportunities arose. The transformation was accidental, unplanned, a side effect of metabolic innovation scaled up over geological time, and yet it was also inevitable. In a sense, because once oxygenic photosynthesis evolved, once it proved more efficient than the alternatives, once it began to spread, the logic of chemistry and planetary biogeochemistry took over, driving the system toward oxygenation whether any organism wanted it or not. Intent is irrelevant in planetary evolution. Only outcomes matter. The rust never stops forming. Even now, in the modern world, iron oxidizes wherever it is exposed to oxygen and moisture. Bridges corrode, cars deteriorate, metal roofs develop patinas of orange and brown. The chemistry is the same as it was 2.4 billion years ago, when the first dissolved iron in the Archean Ocean encountered the first oxygen molecules released by cyanobacteria. Iron atoms lose electrons, oxygen atoms gain them, and ferrous iron transforms into ferric oxide, insoluble and brightly colored. The reaction is spontaneous, thermodynamically favorable, releasing energy that dissipates as heat into the environment. It requires no catalyst, no biological intervention. Though biology originally provided the oxygen that made the reaction possible, rust is a monument to the great oxygenation event, a persistent reminder that the planet's surface chemistry has been permanently altered, that the transition from anoxic to oxic was irreversible, that every breath taken by every aerobic organism depends on a transformation that began in the Paleoproterozoic and has never stopped. The red beds continue to accumulate. Deserts and river valleys and floodplains across the modern continents are stained with iron oxides. Sediments colored in shades of red and orange and ochre by the same weathering processes that began after oxygen appeared in the atmosphere. The Colorado Plateau, the Painted Desert, the red sands of the Australian outback, all owe their colors to oxidized iron, to rust distributed through sandstone and mudstone and conglomerate. The pigmentation is not merely aesthetic, it is diagnostic, a marker of oxidizing conditions, a signal that the sediments were deposited in an oxygen-rich environment. Geologists use the color of sedimentary rocks as a rough proxy for ancient atmospheric chemistry, red beds indicating oxygenated conditions, gray or greenish beds suggesting anoxic or reducing environments. The proxy is imperfect, subject to complications from diagenesis and metamorphism and local redox variability, but it is useful. Easy to assess in the field. A first-order constraint on the oxygenation state of the ancient world. Only took a few decades of stratigraphic mapping to recognize the global distribution of red beds and their temporal appearance in the rock record. The nitrogen cycle has stabilized. Modern nitrogen fixation is dominated by bacteria and archaea, primarily in soils and marine sediments, with some contribution from symbiotic nitrogen fixers in plant roots. The process is energy-intensive, requiring the nitrogenase enzyme complex, which contains molybdenum and iron and is exquisitely sensitive to oxygen. Nitrogenase is poisoned by oxygen, irreversibly inactivated if exposed to O2 concentrations above a low threshold. To protect the enzyme, nitrogen-fixing organisms have evolved various strategies. Some form specialized oxygen-free cells called heterocysts. Others fix nitrogen only at night when photosynthesis has ceased and oxygen levels drop. Still others live in low-oxygen environments like waterlogged soils or anoxic sediments. The adaptations are elegant, effective, allowing nitrogen fixation to persist in an oxygenated world despite the fundamental incompatibility between nitrogenase and oxygen. Only took biochemistry a few decades to work out the mechanisms. The paradox is that oxygen enabled nitrogen fixation on a global scale even as it threatened to shut it down before the Great Oxygenation Event. Molybdenum was scarce in seawater, limiting the activity of molybdenum-dependent nitrogenase. After oxygenation, molybdenum became abundant, weathered from continental rocks and delivered to the ocean, allowing nitrogen fixation to expand and support higher rates of primary productivity. But oxygen also created the need for protective mechanisms, for heterocysts and temporal separation and anoxic microenvironments. The net result is a nitrogen cycle that is more productive but also more complex, more tightly regulated, more dependent on the spatial and temporal partitioning of metabolic processes. Evolution does not optimize for simplicity, it optimizes for functionality, and functional systems are often complicated, layered, full of trade-offs and compromises. The phosphorus cycle remains the ultimate limit. Phosphorus, unlike nitrogen or carbon, has no atmospheric reservoir. It is delivered to the ocean solely through the weathering of phosphate-bearing minerals on land, a slow process limited by the availability of phosphate rock and the rate of chemical weathering. Once in the ocean, phosphorus is rapidly taken up by photosynthetic organisms, incorporated into DNA and RNA and ATP and cell membranes, pass through the food web, and eventually buried in sediments when organisms die and sink. The burial removes phosphorus from the active biogeochemical cycle, sequestering it in sedimentary rocks for hundreds of millions of years until tectonic processes uplift the rocks and weathering releases the phosphorus again. The cycle operates on geological time scales, far slower than the nitrogen or carbon cycles, and it sets a hard limit on long-term primary productivity. No matter how much nitrogen or carbon dioxide is available, if phosphorus is scarce, photosynthesis cannot proceed. Only took the development of nutrient limitation theory in the mid-20th century to recognize phosphorus as the ultimate limiting nutrient in marine systems. The oxygen concentration has fluctuated. Atmospheric oxygen levels did not simply rise to 21% and remain there. They have varied considerably over the Phanerozoic, the last 541 million years, rising as high as 30 to 35% during the Carboniferous and Permian periods and falling as low as 15% during the Triassic and Jurassic. The variations are recorded in charcoal abundances, in the size of fossil insects and other arthropods that rely on passive diffusion for oxygen delivery, in geochemical proxies like the isotopic composition of carbon and sulfur. The causes of the variations are linked to the balance between oxygen sources and sinks. Periods of high oxygen correspond to times when organic carbon burial rates were elevated, locking carbon away and allowing oxygen to accumulate. Periods of low oxygen correspond to times when oxidative weathering or volcanic activity consumed oxygen faster than photosynthesis could produce it. The system is dynamic, responsive to changes in tectonics, climate, biology, and orbital parameters. Only took paleoclimate modeling to show how sensitive oxygen levels are to perturbations. High oxygen levels have consequences. During the Carboniferous, when oxygen concentrations approached 35%, wildfires were more frequent and intense, burning through forests and leaving thick charcoal layers in the sedimentary record. Insects grew larger, some dragonflies reaching wingspans of 70 centimeters, able to support their large bodies because the high oxygen concentration facilitated passive gas exchange through their tracheal systems. The ecosystems were different, structured by oxygen availability in ways that are difficult to observe in the modern world. Low oxygen levels, conversely, stress aerobic organisms, limit their size and activity, shift competitive balances towards species with more efficient respiratory systems or greater tolerance for hypoxia. The Earth's biosphere is not static. It responds to changes in atmospheric composition, evolving, adapting, sometimes thriving and sometimes struggling depending on the oxygen available. The oxygen we breathe is ancient, in a sense. Individual oxygen molecules have short lifetimes, typically reacting within years to decades after being produced, oxidizing organic matter or reduced minerals or combining with other atmospheric gases. But the reservoir is maintained, replenished constantly by photosynthesis, kept at roughly 21% by the balance between production and consumption. The oxygen you inhale with each breath was produced recently, probably within the last few years, by a plant or alga or cyanobacterium somewhere on Earth, but that organism was part of a lineage stretching back billions of years, using metabolic pathways that evolved in the Archean or Proterozoic, participating in biogeochemical cycles that have operated continuously since the Great Oxygenation Event. Breathing connects you to deep time, links your metabolism to the planetary transformation that made complex life possible, makes you, in a very real sense, a participant in the ongoing chemistry of the oxygenated Earth. Only took a few moments of reflection to realize the connection. The poison became indispensable. Oxygen, which caused mass extinctions and climate catastrophes and the collapse of entire ecosystems when it first appeared, is now essential to the vast majority of complex life on Earth. Aerobic metabolism is more efficient, providing more energy per unit of glucose than any anaerobic alternative. That efficiency enables larger bodies, faster movement, complex nervous systems, warm-bloodedness, intelligence. Without oxygen, none of these would be possible. Humans, mammals, birds, fish, insects, all are obligate aerobes, unable to survive without a continuous supply of O2. The dependency is absolute. Remove oxygen from the atmosphere, and complex life collapses within minutes. The irony is that what was once toxic is now necessary. What was once a pollutant is now the basis of the biosphere's energy economy. Evolution does not remember history. It only responds to present conditions, shaping organisms to fit the world as it is, not as it was. Only took a few billion years to go from oxygen as catastrophe to oxygen as requirement. The sediments remember. Cores drilled from the seafloor, columns of mud and clay extracted from beneath kilometers of water. Preserve chemical signals from the past, isotopes of carbon and sulfur, trace element concentrations, the mineralogy of iron and manganese compounds. All record the redox state of the ocean and atmosphere at the time of deposition. The signals are noisy, affected by diagenesis, the post-depositional alteration of sediments as they compact and lithify, as pore waters migrate through them, as minerals dissolve and reprecipitate. But with careful analysis, statistical averaging across many samples, and cross-validation between different proxies, a coherent picture emerges. The ocean was anoxic in the deep layers for most of the Proterozoic. It oxygenated episodically during the Neoproterozoic and fully during the Cambrian. It has fluctuated since then, with intervals of widespread ocean anoxia during mass extinction events, but it has never returned to the persistently stratified state that characterized the Proterozoic. The sediments are a library, an archive written in chemistry, accessible to anyone with the tools to read it. Only took the development of isotope ratio mass spectrometry to unlock the archive. Ice cores provide a different archive. Air bubbles trapped in Antarctic and Greenland ice sheets preserve samples of the atmosphere from the past 800,000 years, allowing direct measurement of oxygen, carbon dioxide, methane, and other gases. The record shows that oxygen concentrations have remained stable at around 21% throughout this interval, varying by less than 1% even during glacial-interglacial cycles, when carbon dioxide fluctuated by nearly a factor of 2. The stability is striking, suggesting strong regulatory feedbacks that buffer oxygen against perturbations. The feedbacks are thought to involve the combustion of organic matter. When oxygen rises, wildfires increase, burning vegetation and consuming oxygen. When oxygen falls, fires decrease, allowing oxygen to accumulate. The feedback is not perfect. It operates on timescales of thousands to tens of thousands of years, but it is effective, maintaining oxygen within a narrow range suitable for aerobic life. Only took a few glacial cycles recorded in ice to demonstrate the stability. But ice cores only extend back 800,000 years, a tiny fraction of Earth's history. For older intervals, indirect proxies are necessary. Charcoal in sedimentary rocks indicates that oxygen concentrations were above the flammability threshold, roughly 13 to 15% of atmospheric composition, because below that threshold, fires cannot spread. The first widespread charcoal deposits appear in the Silurian and Devonian periods, around 420 to 360 million years ago, suggesting that oxygen reached modern levels, or close to it. By this time, earlier intervals lack charcoal, implying lower oxygen concentrations, though the absence of charcoal could also reflect the absence of terrestrial vegetation rather than low oxygen. Interpreting the proxy requires caution, attention to context, consideration of alternative explanations. Only took several decades of paleobotany to disentangle the signals. Insects provide another line of evidence. Giant insects, particularly dragonflies and millipedes, are found in Carboniferous and Permian fossils, dating to around 300 million years ago. Some dragonflies had wingspans exceeding 70 centimeters, far larger than any modern dragonfly, which rarely exceed 15 centimeters. The size is difficult to explain under modern oxygen concentrations because insect respiratory systems rely on passive diffusion through tracheal tubes, and diffusion is too slow to supply oxygen to tissues in very large bodies. But if atmospheric oxygen were higher, diffusion would be faster, allowing larger body sizes. Modeling suggests that oxygen concentrations of 30 to 35% would be sufficient to support the observed sizes of Carboniferous insects. The correspondence between modeled oxygen levels and other proxies, like carbon isotopes and charcoal abundances, strengthens the inference that oxygen was indeed elevated during this interval. Only took biomechanical modeling of insect respiration to connect physiology to atmospheric chemistry. The coal deposits tell the same story. The Carboniferous period is named for the extensive coal beds that formed during this time. Thick accumulations of partially decomposed plant material buried in swamps and peat bogs and eventually compressed into coal. The high rates of organic carbon burial imply that decomposition was suppressed. That plant material accumulated faster than bacteria and fungi could break it down. One explanation is that the organisms capable of decomposing lignin and cellulose, the structural polymers in wood, had not yet fully evolved. Another is that waterlogged, anoxic conditions in the swamps inhibited aerobic decomposition. A third is that high oxygen concentrations in the atmosphere promoted rapid plant growth, overwhelming the capacity of decomposers. All three factors probably contributed. The result was the removal of vast amounts of carbon from the active biogeochemical cycle, burial in sediments that would eventually become coal, and a corresponding rise in atmospheric oxygen as the carbon was sequestered and the oxygen was left behind. Only took industrial-scale coal mining to reveal the extent of Carboniferous carbon burial. The oxygen rise had ecological consequences. Forests expanded, spreading across continents, covering swamps and floodplains and upland areas. The forests were dominated by lycophytes, tree-like plants related to modern club mosses, along with early conifers and ferns. The canopy was dense, the productivity high, and the ecosystem supported diverse arthropod communities, including millipedes, scorpions, spiders, and the giant dragonflies. Vertebrates colonized the land, evolving from lobe-finned fish into amphibians and eventually into early reptiles. The Carboniferous was a time of rapid terrestrial diversification, the biosphere expanding into habitats that had been barren or sparsely colonized in earlier periods. The expansion was enabled, in part, by the high oxygen concentrations, which supported larger body sizes and more active metabolisms in both plants and animals. Only took paleontology a century to map the Carboniferous ecosystems, but high oxygen also made the world more flammable. Wildfires were frequent, spreading through forests, consuming vegetation, leaving charcoal layers that are abundant in Carboniferous sediments. Some charcoal beds are meters thick, recording fires that burned for extended periods, possibly years, across vast areas. The fires were part of the carbon cycle, oxidizing plant material back into carbon dioxide, returning carbon to the atmosphere that would otherwise have been buried. They also affected forest structure, favoring fire-resistant species, creating mosaics of burned and unburned areas, shaping ecosystems in ways analogous to modern fire-adapted landscapes like the African savanna or the boreal forests of North America. Fire is a natural process, but its frequency and intensity are modulated by oxygen availability. The Carboniferous was a world on fire, more so than any subsequent period. Only took fire ecology a few decades to recognize the role of oxygen in wildfire dynamics. The oxygen eventually declined. By the end of the Permian period, around 250 million years ago, oxygen concentrations had dropped back toward modern levels, possibly falling below 20% during the Triassic as carbon burial rates decreased and oxidative weathering increased. The decline coincided with the end Permian mass extinction, the most severe biodiversity crisis in the fossil record, when more than 90% of marine species and 70% of terrestrial vertebrate species went extinct. The causes of the extinction are complex, involving massive volcanic eruptions in Siberia, ocean acidification, anoxia, global warming, and possibly the release of methane from seafloor deposits. The drop in oxygen may have contributed, stressing aerobic organisms, reducing their tolerance to other environmental perturbations, amplifying the effects of warming and anoxia. The extinction reset the biosphere, eliminating dominant lineages and opening ecological opportunities that would be filled slowly during the Triassic and Jurassic by new groups. Dinosaurs, mammals, marine reptiles, modern coral reefs only took a few million years to recover diversity. Though the composition of ecosystems was permanently altered, the oxygen level stabilized again. Through the Mesozoic and Cenozoic, atmospheric oxygen has remained close to 21%, fluctuating within a few percentage points, but never returning to the extremes of the Carboniferous or the lows of the Triassic. The stability reflects a mature biogeochemical system, one in which the feedbacks between oxygen production, carbon burial, weathering, and combustion are well balanced, capable of responding to perturbations without runaway excursions in either direction. The system is not static, but it is resilient, buffered by the size of the atmospheric reservoir, by the diversity of organisms contributing to oxygen production and consumption, by the slow pace of geological processes that modulate the long-term balance. Resilience, in Earth system science, is a measure of how much disturbance a system can absorb before shifting to a different state. The oxygen cycle has proven resilient for hundreds of millions of years. Only took global change biology to appreciate the importance of resilience in maintaining habitability. Humans are altering the system. Combustion of fossil fuels, coal, oil, natural gas, is oxidizing carbon that was buried tens to hundreds of millions of years ago, returning it to the atmosphere as carbon dioxide and consuming oxygen in the process. The oxygen consumption is measurable but small relative to the atmospheric reservoir. Burning all known fossil fuel reserves would reduce atmospheric oxygen by less than 1%. A negligible change given that oxygen is 21% of the atmosphere and the reservoir is enormous. The effect on oxygen levels is trivial. The effect on carbon dioxide levels is not. CO2 concentrations have risen from roughly 280 parts per million before the Industrial Revolution to over 420 parts per million today, driving global warming, ocean acidification, shifts in precipitation patterns, and ecological disruptions. The perturbation is rapid, faster than anything in the geological record except for asteroid impacts and large igneous province eruptions. The biosphere is responding, adapting where possible, but the pace of change is challenging and the outcomes are uncertain. Only took a few decades of climate science to recognize the scale of the disruption. The oxygen we produce is also being consumed. Deforestation, conversion of forests and grasslands to agriculture, degradation of marine ecosystems, all reduce the global capacity for photosynthesis, lowering the rate at which oxygen is produced. At the same time, human activities increase oxygen consumption through combustion, respiration by livestock and human populations, and oxidation of organic waste. The net effect on atmospheric oxygen is still small, but the trajectory is concerning if it continues. More worrying is the loss of photosynthetic capacity itself, the reduction in the biosphere's ability to regulate atmospheric composition, to buffer against perturbations, to maintain the conditions that support complex life. The oxygen cycle depends on living organisms, and those organisms depend on habitats, on stable climates, on nutrient availability, on the absence of toxins and pollutants, disrupt the organisms, and the cycle falters. Only took a few decades of ecological research to demonstrate the fragility of ecosystem services. The cyanobacteria, still photosynthesizing, still producing oxygen, are threatened by ocean warming, acidification, nutrient pollution, changes in ocean circulation. In some regions, warming promotes cyanobacterial blooms, creating oxygen-depleted dead zones when the blooms die and decompose. In other regions, stratification intensifies, reducing nutrient upwelling, limiting primary productivity. The ocean's role in oxygen production is smaller than the terrestrial biospheres. Phytoplankton produce perhaps half of global oxygen, but it is not negligible. And changes in ocean productivity have cascading effects on marine food webs, on fisheries, on the biogeochemical cycles that regulate atmospheric composition. The ocean is not a passive reservoir. It is an active participant in the Earth system, responding to changes, feeding back, sometimes stabilizing and sometimes amplifying. Only took oceanography a century to map the ocean's role in global cycles. The future of oxygen is secure. For now. The atmospheric reservoir is large enough that even dramatic changes in production or consumption rates would take centuries to millennia to significantly alter oxygen concentrations. Humans could burn all the fossil fuels, cut down all the forests, and atmospheric oxygen would still remain above 15%, still sufficient to support aerobic life. The oxygen cycle is buffered by its size, by the slow pace of geological processes, by the resilience of the biosphere. But that security is not guaranteed indefinitely. On timescales of millions of years, continued disruption of the carbon cycle, alteration of weathering rates, changes in tectonic activity could shift the system into a new state, one with lower oxygen, reduced productivity, impoverished ecosystems. The planet has been there before, during the Proterozoic, and it could return there if the conditions align. Only took Earth system modeling to show that multiple stable states are possible, that the modern oxygenated world is not the only equilibrium. The rocks at Mistaken Point, Newfoundland, Preserve a Moment, the outcrop, exposed along a wave-cut platform on the coast, contains fossils of ediacaran organisms, impressions of frond-like forms pressed into volcanic ash that fell into the sea roughly 565 million years ago. The ash buried the organisms rapidly, preserving them in exquisite detail, recording their positions on the seafloor, their orientations relative to currents, their community structure. The fossils are not bones or shells, but compression fossils, the shapes of soft bodies flattened into 2 dimensions, visible only because the ash was fine-grained enough to capture their outlines. The assemblage is dense, with hundreds of individuals per square meter, suggesting that these organisms were abundant, perhaps dominant in their ecosystem. They are also strange, unlike any modern life, with fractal branching patterns and radial symmetries that do not fit easily into known phylogenetic categories. The site is a window into the aftermath of the Gasquias glaciation, one of the Neo-Proterozoic snowball Earth events, which ended around 580 million years ago. The glaciation was severe, ice sheets advancing to low latitudes, ocean surface temperatures dropping to near freezing. When the ice melted, the ocean oxygenated, nutrients flooded from weathering of exposed continents, and photosynthetic productivity surged. The ediacaran organisms diversified in this post-glacial world, exploiting the high oxygen concentrations. The nutrient availability, the ecological opportunities opened by the environmental transformation. They were not the first multicellular organisms, but they were among the first to achieve large size, to form complex communities, to leave fossils abundant enough and distinctive enough to be recognized globally. Only took several decades of fieldwork across multiple continents to map the distribution of ediacaran assemblages. The fronds were probably sessile, anchored to the seafloor, unable to move. Some may have filter-fed, extracting organic particles from seawater using cilia or sticky mucus. Others may have been osmotrophs, absorbing dissolved organic matter directly through their surfaces. A few may have hosted photosynthetic symbionts, much like modern corals host zooxanthellae, using the symbionts' photosynthesis to supplement their nutrition. The metabolic strategies are uncertain, inferred from morphology and environmental context rather than from direct evidence, because soft tissues do not preserve biochemical signatures after 565 million years of diagenesis and metamorphism. The interpretations are plausible but speculative. Models rather than facts. Subject to revision as new data emerge. Only took humility to admit that much about the Ediacara biota remains unknown. Some Ediacaran organisms were mobile. Trace fossils from this interval include horizontal burrows, shallow trails left by organisms moving across the sea floor or just beneath the sediment surface. The burrows are simple, lacking the complexity of later trace fossils, but they indicate the presence of animals capable of coordinated movement, of sensing their environment, of responding to chemical or tactile cues. The animals were probably bilaterians, worm-like forms with muscles and a hydrostatic skeleton, propelling themselves through sediment by peristaltic contractions. The trace fossils become more abundant and more complex toward the end of the Ediacaran, suggesting that burrowing behavior was evolving, diversifying, becoming more widespread as oxygen levels rose and the energetic costs of active movement became affordable. Only took ichnology, the study of trace fossils, to recognize the behavioral innovations recorded in ancient sediments. The transition to the Cambrian is marked by predation. The earliest predators appear in the late Ediacaran and early Cambrian, small arthropods and worm-like forms with grasping appendages, jaws, or other structures capable of capturing and consuming other organisms. Predation changes everything. It creates selective pressure for defensive adaptations, shells, spines, mineralized skeletons, burrowing deeper into sediment, faster locomotion, camouflage. It also creates selective pressure for offensive adaptations, better sensory systems, faster reflexes, stronger muscles, venom, or other chemical weapons. The result is an evolutionary arms race, a feedback loop in which predators and prey co-evolve, each driving the other toward greater complexity and specialization. The Cambrian explosion is, in part, the fossil record of this arms race, the sudden appearance of hard parts and complex morphologies reflecting the intensity of selection in a world where being eaten became a significant mortality factor. Only took evolutionary ecology several decades to work out the dynamics of predator-prey coevolution. The first shells were probably defensive. Mineralized skeletons are costly to produce, requiring energy and resources to extract calcium or silica from seawater and deposit it in organized structures. The cost must be offset by a benefit, and the most obvious benefit is protection from predation. Small shelly fossils, millimeter-scale shells and sclerites, appear in the earliest Cambrian, around 540 to 530 million years ago, distributed globally, representing a diversity of organisms including mollusks, brachiopods, and various extinct groups. The shells are often fragmentary, isolated pieces of armor that covered parts of the body but did not form complete enclosures. Later, shells become more elaborate, fully enclosing the soft tissues, hinged or coiled or segmented in ways that allow for growth while maintaining protection. The evolution of shells is iterative, experimental, exploring morphospace through trial and error, with natural selection winnowing the failures and amplifying the successes. Only took comparative anatomy and functional morphology to understand the biomechanics of shell design. Eyes evolve. The first compound eyes, with multiple lenses and photoreceptor cells arranged to form images, appear in trilobites and other arthropods in the early Cambrian. Eyes are complex organs, requiring precise spatial organization of tissues, sophisticated neural processing of visual information, and coordination between the eye and the rest of the nervous system. The evolution of eyes represents a major increase in cognitive capability, allowing organisms to detect predators from a distance, to locate prey, to navigate through complex environments, to assess potential mates. Vision changes behavior. It enables active hunting rather than passive filter feeding or scavenging. It enables escape responses, fleeing from threats rather than relying solely on armor or toxins. It enables social interactions, communication through visual signals, coordination in groups. The behavioral innovations enabled by vision are as important as the morphological innovations in shells and appendages. Only took neurobiology and sensory ecology to appreciate the cognitive implications of eyes. The oxygen requirement for eyes and brains is substantial. Neural tissue is metabolically expensive, consuming ATP at high rates to maintain ion gradients across cell membranes, to synthesize neurotransmitters, to support action potentials and synaptic transmission. Large brains or complex nervous systems require correspondingly large energy supplies, which in turn require efficient aerobic metabolism and high oxygen availability. The Cambrian animals, with their compound eyes and centralized nervous systems and active behaviors, could not have evolved without the elevated oxygen concentrations that characterized the Neoproterozoic and Cambrian. The correlation between oxygen levels and the appearance of complex animals is not coincidental, it is causal, a direct consequence of the energetic demands of neural tissue and the efficiency of aerobic respiration in meeting those demands. Only took bioenergetics to quantify the oxygen requirements for different tissue types. The Cambrian seafloor was a busy place. Trilobites scuttled across sediments, their compound eyes scanning for food or predators. Anomalocaridids, large predatory arthropods with grasping appendages, hunted smaller animals in the water column. Priapulid worms burrowed through sediment, consuming organic matter and leaving networks of vertical burrows. Brachiopods and mollusks attached to hard substrates, filtering plankton from seawater. Echinoderms grazed on microbial mats or detritus. The diversity of body plans, of ecological roles, of behaviors was unprecedented, unmatched in the preceding 4 billion years of life on Earth. The Cambrian represents the transition from a microbial world dominated by bacteria and unicellular eukaryotes to a macroscopic world dominated by animals with complex organs and behaviors and interactions. The transition was rapid, geologically speaking, compressed into a few tens of millions of years, but it was built on billions of years of prior evolution, of metabolic innovations and genetic toolkits and environmental transformations that set the stage. Only took the integration of paleontology, geochemistry, and molecular biology to reconstruct the full arc of the transition. The oxygen sinks never disappear entirely. Even in the modern oxygenated world, anoxic environments persist. The deep Black Sea, isolated from circulation and rich in hydrogen sulfide, the sediments of wetlands and rice paddies, waterlogged and devoid of oxygen, the guts of termites and ruminants, hosting methanogenic archaea that produce methane, the oxygen minimum zones in the ocean, layers at midwater depths where oxygen consumption exceeds supply. These environments are refugia for anaerobic organisms, pockets where the chemistry of the Archean persists, where metabolisms that evolved billions of years ago continue to operate. The organisms in these environments are not primitive. They have evolved continuously since the Archean, adapting to changing conditions, optimizing their metabolisms, but they employ ancient biochemical pathways, strategies for extracting energy without oxygen that predate the Great Oxygenation Event. Methanogens, in particular, remain abundant and ecologically significant. They produce roughly 500 million tons of methane annually, released into the atmosphere from wetlands, from livestock, from thawing permafrost, from seafloor sediments. The methane is a potent greenhouse gas, molecule for molecule far more effective at trapping heat than carbon dioxide, though its atmospheric lifetime is shorter, only about a decade before it is oxidized into CO2 and water. The methanogen-produced methane contributes to global warming, amplifies climate feedbacks, complicates efforts to predict and manage atmospheric composition. The organisms are invisible, microscopic, often ignored in discussions of climate, but their collective metabolic output is planetary scale, significant enough to affect radiative forcing and temperature. Only took atmospheric chemistry several decades to quantify the contribution of biological methane sources. Sulfate-reducing bacteria also persist. In marine sediments, they consume organic matter using sulfate as an electron acceptor, producing hydrogen sulfide as a byproduct. The sulfide diffuses upward through the sediment column, reacting with iron to form pyrite, or escaping into the overlying water where it can be toxic to aerobic organisms. In some coastal regions, sulfate reduction is intense enough to create oxygen-depleted zones, contributing to hypoxia and fish kills. The process is natural, part of the sulfur cycle, but it is sensitive to nutrient loading from agriculture and wastewater, which increases organic matter input to sediments and stimulates bacterial activity. Human perturbations amplify natural processes, pushing systems towards states that are less favorable for aerobic life. Only took marine biogeochemistry a few decades to link nutrient pollution to hypoxia and dead zones. Iron oxidation continues, both biologically and abiotically. Iron-oxidizing bacteria, which gain energy by catalyzing the oxidation of ferrous to ferric iron, are found in acidic mine drainage, in hydrothermal vents, in groundwater seeping through iron-rich soils. The bacteria accelerate a reaction that would occur anyway in the presence of oxygen, but they couple it to metabolism, harvesting the small energy yield and using it to fix carbon dioxide, to synthesize cellular components, to reproduce. The organisms are extremophiles in many cases, tolerating low pH, high metal concentrations, temperatures near boiling. They are also ancient, their metabolic pathways likely dating to the Archean, adapted over billions of years to extracting energy from iron in a world that has transitioned from anoxic to oxic. Only took microbial ecology a few decades to culture and characterize iron-oxidizing bacteria from diverse environments. The metabolic diversity of microbes is staggering. Bacteria and archaea have evolved to exploit nearly every chemical disequilibrium on Earth, every gradient in redox potential, every pair of electron donors and acceptors that can yield energy when reacted. They oxidize hydrogen, sulfur, iron, manganese, ammonia, methane. They reduce oxygen, nitrate, sulfate, carbon dioxide, ferric iron. They ferment sugars, amino acids, complex organic polymers. They photosynthesize using chlorophyll, bacteriochlorophyll, rhodopsin. They fix nitrogen, fix carbon, break down cellulose, produce toxins, form biofilms, survive radiation, endure freezing, tolerate salt concentrations that would desiccate any eukaryotic cell. The metabolic repertoire of microbes far exceeds that of plants and animals, which rely on a narrow suite of pathways: aerobic respiration, photosynthesis, fermentation. Microbes are the metabolic foundation of the biosphere, the engines driving biogeochemical cycles, the organisms that maintain the habitability of the planet. Only took microbiology a century to catalog a fraction of microbial diversity. Eukaryotes, by comparison, are metabolically conservative. Nearly all eukaryotes rely on mitochondria for aerobic respiration, and those that have lost mitochondria or modified them into hydrogenosomes or mitosomes are rare. Restricted to specific anaerobic environments, nearly all photosynthetic eukaryotes rely on chloroplasts derived from cyanobacteria, with a few exceptions like dinoflagellates that have acquired plastids through multiple rounds of endosymbiosis. The metabolic uniformity reflects the evolutionary history of eukaryotes. They originated through endosymbiosis, incorporating bacterial metabolic machinery, and they have been largely conservative since then, optimizing and diversifying within the constraints imposed by their bacterial-derived organelles. The success of eukaryotes is not metabolic innovation, but morphological and behavioral complexity, made possible by the energy provided by mitochondria. Only took cell biology several decades to appreciate the centrality of organelles to eukaryotic evolution. The division between prokaryotes and eukaryotes is not absolute. Lateral gene transfer, the movement of genes between distantly related organisms, blurs the boundaries. Bacteria acquire genes from archaea and vice versa. Eukaryotes acquire genes from bacteria. sometimes through endosymbiosis, but also through viral vectors or direct uptake of DNA from the environment. The result is a mosaic genome, a patchwork of genes with different evolutionary histories, complicating phylogenetic reconstruction and challenging simplistic narratives of descent with modification. Evolution is not a tree, but a network, with branches that fuse and exchange genetic material, with lineages that hybridize and merge. The network is densest among microbes, where lateral gene transfer is frequent, but it extends to eukaryotes as well, particularly in their organella genomes. Only took comparative genomics to reveal the extent of lateral gene transfer and its role in evolution. The origin of eukaryotes remains partially mysterious. The endosymbiosis that gave rise to mitochondria is well established, supported by genomic data, phylogenetic analyses, and the presence of double membranes around mitochondria that reflect their bacterial origin. But the identity of the host cell, the circumstances of the initial engulfment, the sequence of events leading to full integration, these are still debated. Recent discoveries of the Asgard archaea, a group that shares many genes with eukaryotes and appears to be the closest living relatives of the eukaryotic lineage, have clarified some aspects of the origin, suggesting that the host was an archaeon rather than a bacterium. But the details remain elusive, obscured by the vast timescale, by the lack of intermediate forms in the fossil record, by the genomic complexity that makes phylogenetic reconstruction difficult. Only took metagenomics and single-cell sequencing to identify the Asgard archaea and place them in the tree of life. The mitochondrion, once free-living, is now imprisoned. It has lost most of its genome, transferring genes to the host nucleus, retaining only a few dozen genes essential for oxidative phosphorylation and that must be tightly regulated in response to the mitochondrion's internal state. The remaining genes are under strong selection, highly conserved across eukaryotic lineages, mutating slowly because most mutations are deleterious. The mitochondrial genome is a molecular fossil, a remnant of the alpha proteobacterium that was engulfed 2 billion years ago, reshaped by billions of years of evolution inside the host cell. It cannot leave. It cannot survive independently. It is fully integrated, obligately symbiotic, its fate tied to the fate of the host. The irreversibility is total. Only took molecular biology several decades to sequence mitochondrial genomes and compare them to free-living bacteria. The continents continue to drift. Plate tectonics, driven by convection in the mantle and the cooling of the planet's interior, moves the continents across the surface at rates of a few centimeters per year. Over millions of years, the movement is substantial. Continents collide, forming mountain ranges. They rift apart, opening ocean basins. They subduct, one plate sliding beneath another, recycling crustal material back into the mantle. The process is continuous, operating since at least the Archean, possibly earlier, reshaping the surface configuration, redistributing land and sea, altering ocean circulation and climate patterns. The movement is slow by human standards, but geologically inexorable, a fundamental aspect of how the planet operates. The current configuration is temporary. The Atlantic Ocean, which began opening around 180 million years ago when the supercontinent Pangaea rifted apart, is still widening, growing by a few centimeters each year as new oceanic crust forms at the mid-Atlantic ridge. The Pacific Ocean is shrinking, its margins consumed by subduction zones, where oceanic plates dive beneath continental plates, generating earthquakes and volcanic arcs. In another 200 to 250 million years, the continents will collide again, forming a new supercontinent, sometimes called Pangaea Proxima or Amasia, depending on the model. The cycle of supercontinent assembly and breakup operates on timescales of 400 to 600 million years, a rhythm that has persisted through much of Earth's history. influencing climate, ocean chemistry, biological evolution. Only took plate tectonic theory several decades to develop predictive models of future continental configurations. The supercontinent cycle affects oxygen levels. When continents are dispersed, weathering occurs over a large geographic area, with diverse climates and topographies, promoting efficient nutrient delivery to the ocean and supporting high primary productivity. When continents are assembled into a supercontinent, the interior becomes arid. Weathering rates decline and nutrient fluxes to the ocean decrease. The reduced productivity lowers oxygen production, and atmospheric oxygen concentrations may decline. Conversely, the breakup of a supercontinent often involves extensive volcanism, releasing carbon dioxide, warming the climate, accelerating weathering, increasing nutrient delivery, and boosting oxygen production. The correlations are not perfect. Other factors like orbital variations, biological innovations, and asteroid impacts also affect oxygen levels, but the supercontinent cycle is a significant driver of long-term oxygen variability. Only took integrated Earth system models to explore the connections between tectonics, climate, and atmospheric composition. Life responds to tectonics. Mountain building creates new habitats, isolated by elevation and climate, promoting speciation. The opening of ocean basins creates barriers to dispersal, fragmenting populations, allowing divergent evolution. The collision of continents brings previously isolated biotas into contact, triggering competition, extinction, and ecological reorganization. The fossil record shows pulses of diversification and extinction correlated with tectonic events. Though the correlations are noisy and the causality is often ambiguous, evolution is opportunistic, responding to environmental change in ways that are contingent on the lineages present. The ecological interactions, the availability of resources, tectonics provides the stage, but biology writes the script. Only took paleobiogeography several decades to map the connections between continental drift and biodiversity patterns. Oxygen is a gift from the past. Every breath taken by an aerobic organism, every molecule of O2 inhaled into lungs or dissolved across gills or diffused into cells, is a gift from cyanobacteria that lived billions of years ago, from the slow accumulation of oxygen over geological time, from the irreversible transformation of the planet's surface chemistry. The oxygen is maintained by ongoing photosynthesis, by plants and algae and cyanobacteria that continue to split water and release O2. But the existence of an oxygenated world, the fact that aerobic respiration is possible, the entire architecture of the modern biosphere, all trace back to the great oxygenation event, to the moment when oxygen began to accumulate and could not be stopped. The transformation was not planned. It was not foreseen. It was the cumulative result of metabolic activity scaled up over planetary dimensions and geological time. Only took billions of years and trillions of organisms to oxygenate a planet. The irony remains. The organisms that oxygenated the world did not benefit, at least not initially. The Archean anaerobes that dominated before oxygenation were driven to extinction or to marginal anoxic refugia. The cyanobacteria themselves had to evolve mechanisms to tolerate oxygen, to detoxify reactive oxygen species, to prevent damage to their cellular machinery. The benefit of oxygen. The energy surplus provided by aerobic respiration only became apparent later, after organisms evolved mitochondria, after eukaryotes diversified, after complex multicellular life emerged. The transformation was a catastrophe that became a prerequisite, a pollution event that became the foundation for everything that followed. Evolution does not optimize for long-term outcomes. It responds to immediate conditions, selecting for traits that work now, indifferent to the distant consequences. Only took evolutionary biology a century to articulate this principle clearly. The time scales are humbling. The Great Oxygenation Event unfolded over tens to hundreds of millions of years, a duration so long that it is difficult to conceptualize. Human civilization has existed for a few thousand years. Recorded history spans a few millennia. The entire Holocene epoch, the interval since the last glacial maximum, is only about 12,000 years old. The time required for oxygen to accumulate in the atmosphere is thousands of times longer than all of human history, longer than the existence of the genus Homo, longer than the evolution of primates, longer than the reign of the dinosaurs. The event is invisible to any observer operating on human timescales. It can only be detected through the integration of geochemical data across millions of years, through the patient accumulation of measurements and the construction of models that span geological epochs. Only took the development of geochronology and isotope geochemistry to make the timescales accessible to analysis. The patience of geology is infinite. Rocks wait. Isotopes decay at fixed rates, indifferent to temperature, pressure, or the passage of time. Sediments accumulate grain by grain, layer by layer, building sequences that record millions of years of history. Minerals crystallize, lock in chemical signatures, preserve information about the conditions of their formation. The information is there, encoded in the rock record, waiting to be read by anyone with the tools and the persistence to extract it. The reading is difficult, requires careful sampling, precise measurement, thoughtful interpretation, but it is possible. The past is not lost, it is preserved, fragmentarily, incompletely, but legibly in the stones themselves. Only took geologists several centuries to develop the techniques for reading the record. The story is unfinished. The Earth is still evolving, still changing, still responding to the interplay of tectonics, climate, biology, and chemistry. Oxygen levels are currently stable, but they will not remain so indefinitely. On timescales of hundreds of millions to billions of years, the sun will continue to brighten. Temperatures will rise, silicate weathering will accelerate, carbon dioxide will be drawn down to levels too low to support photosynthesis, and oxygen production will cease. The atmosphere will gradually lose its oxygen, consumed by weathering, by volcanic gases, by oxidation of organic matter. The planet will return, eventually, to an anoxic state, though the timeline is uncertain. Dependent on factors that are difficult to predict, the oxygenated world is a phase, not a permanent condition, a transient state in the long evolution of the Earth. Only took planetary science a few decades to model the far future evolution of the atmosphere. The sky, as it exists now, is a collaboration. The blue color comes from Rayleigh scattering, the preferential scattering of shorter wavelengths of light by nitrogen and oxygen molecules in the atmosphere. Without oxygen, without nitrogen, the sky would appear differently, perhaps pale or white or tinged with other colors depending on the atmospheric composition. The blue is incidental, a side effect of molecular properties and the wavelengths of sunlight, but it is also diagnostic, a visible reminder that the atmosphere contains gases that scatter light in specific ways. The color is fragile, sensitive to changes in composition, in particulate loading, in the thickness of the atmosphere. On Mars, with its thin carbon dioxide atmosphere and dust-laden air, the sky appears butterscotch or pinkish. On Titan, Saturn's largest moon, with its thick nitrogen-methane atmosphere, the sky is orange, tinted by organic haze. The blue of Earth's sky is unique in the solar system, a consequence of the specific mixture of gases that accumulated over billions of years of geological and biological evolution. Only took atmospheric optics a century to explain why the sky is blue. Clouds form, dissipate, reform. Water vapor rises from the ocean, condenses into droplets when it cools, falls as rain, evaporates again, cycles endlessly through the hydrological system. The cycle is powered by solar energy, by the sun's heat that drives evaporation and atmospheric circulation. It is also modulated by biology. Plants transpire water vapor through their leaves, contributing to atmospheric moisture. Phytoplankton release dimethyl sulfide, which oxidizes into sulfate aerosols that seed cloud formation. The connections between biology and climate are multifaceted, operating through feedbacks that are sometimes stabilizing and sometimes destabilizing. Biology does not control climate, but it influences it, nudges it, participates in the regulation of temperature and moisture and atmospheric composition. Only took Earth system science several decades to quantify the biological contributions to cloud formation and precipitation. The nitrogen cycle is also biological. Nitrogen fixation, the conversion of atmospheric N2 into ammonia, is performed almost exclusively by bacteria and archaea, with some contribution from industrial processes like the Haber-Bosch synthesis of fertilizers. The fixed nitrogen is assimilated into proteins and nucleic acids, passed through food webs, eventually returned to the atmosphere by denitrifying bacteria that reduce nitrate back to N2. The cycle is closed, balanced over long time scales, though human activities have perturbed it significantly by producing synthetic fertilizers and by cultivating nitrogen-fixing crops. The perturbation has doubled the rate of nitrogen fixation globally, increased nutrient loading to rivers and coastal waters, contributed to eutrophication and hypoxia. The nitrogen cycle, like the carbon and oxygen cycles, is sensitive to human influence, responsive to changes in land use, agriculture, combustion. Only took a few decades of biogeochemical research to map the human perturbations to the nitrogen cycle. Phosphorus, by contrast, has no atmospheric component. It cycles through weathering, biological uptake, sedimentation, uplift, and weathering again, operating entirely in the lithosphere, hydrosphere, and biosphere. The cycle is slow, limited by weathering rates, and it has been significantly perturbed by mining of phosphate rock for fertilizers. The mined phosphorus is applied to agricultural fields, where much of it runs off into rivers and eventually the ocean, accumulating in coastal sediments rather than being recycled through uplift and weathering. The perturbation is unsustainable on long time scales, depleting phosphate reserves and shifting phosphorus distribution in ways that may affect future agricultural productivity. The phosphorus cycle, unlike carbon or nitrogen, cannot be replenished from the atmosphere or by biological processes. It depends on geology, on the slow pace of tectonic uplift and erosion. Only took agricultural science several decades to recognize the non-renewable nature of phosphate resources. The elements cycle at different rates. Carbon cycles through the atmosphere, ocean and biosphere on time scales of years to thousands of years, with a slower component cycling through carbonate rocks over millions of years. Nitrogen cycles on time scales of decades to centuries, depending on the pathway. Oxygen, maintained by photosynthesis and consumed by respiration and weathering, has a residence time in the atmosphere of millions of years, long enough to buffer short-term perturbations, but short enough to respond to major changes in biological productivity or geological activity. Phosphorus cycles on time scales of tens of thousands to millions of years, limited by weathering. The different rates reflect the different properties of the elements, their chemical behavior, their biological importance, their abundance. The cycles interact, coupled through biology and chemistry, creating feedbacks that stabilize or destabilize the Earth system depending on the context. Only took systems ecology several decades to model the coupling between elemental cycles. The deep time perspective reveals patterns. Oxygen levels have risen, fallen, risen again over Earth's history, responding to changes in photosynthetic productivity, continental weathering, volcanic activity. Biological innovations. The trajectory is not linear, not progressive, not teleological. It is contingent, path-dependent, shaped by events that could have gone differently. If cyanobacteria had not evolved oxygenic photosynthesis, oxygen would not have accumulated. If the Huronian glaciations had been more severe, life might have been extinguished before eukaryotes evolved. If the Neoproterozoic oxygenation had not occurred, the Cambrian explosion might have been delayed or might never have happened. The history is full of branching points, moments when the trajectory could have diverged, leading to a different planet, a different biosphere, a different distribution of life and chemistry. The actual history is one realization among many possible histories, selected not by design but by the contingencies of geology and biology and chance. Only took paleontology and Earth system science several decades to appreciate the contingency inherent in planetary evolution. The oxygen in the air is young. Individual molecules persist for only a few years before being consumed by respiration or oxidation, replaced by new molecules from photosynthesis. The turnover is rapid, the reservoir constantly refreshed, but the existence of the reservoir itself, the fact that oxygen comprises 21% of the atmosphere rather than 0%, is ancient, dating back to the Great Oxygenation Event, maintained by the ongoing activity of photosynthetic organisms. The distinction matters. The molecules are ephemeral, but the state of the system is persistent, stabilized by feedbacks, buffered by the size of the reservoir, resilient to perturbations. Systems have memory even when their components are transient. Only took dynamical systems theory to formalize the distinction between state variables and flow variables. The silence is unbroken. There is no sound in deep time. No audio recording of the Archean Ocean or the Proterozoic wind or the Cambrian seafloor. There is only the rock record, silent and still, preserving chemistry and morphology but not sound, not color, not motion. The reconstruction is necessarily incomplete. A translation from the language of isotopes and minerals into the language of narrative. A projection of understanding onto events that occurred billions of years ago and left only fragmentary traces. The reconstruction is informed by physics, by chemistry, by biology, by the assumption that natural laws operated in the past as they do now. But it is also imaginative, filling gaps where data are absent, interpolating between measurements, constructing coherence from fragments. The result is a story, grounded in evidence, but also shaped by the choices of what to emphasize, what to omit, what to interpret as significant. Only took the philosophy of science several decades to articulate the theory-laden nature of observation and interpretation. The cyanobacteria persist, unchanged in essentials, adapted in details. Modern Synechococcus and Prochlorococcus, abundant in the open ocean. Photosynthesize much as their ancestors did 2 billion years ago, using the same photosystems, the same enzymes, the same basic metabolic pathways. The genetic sequences have diverged, accumulated mutations, been shaped by selection, but the core machinery remains recognizable, homologous to the ancient forms. Evolution conserves what works, modifies at the margins, experiments with variations while retaining the fundamental architecture. The cyanobacteria are living fossils, biochemically speaking, lineages that have persisted across geological epochs, survived mass extinctions, adapted to changing oceans and atmospheres without losing their essential identity. Only took molecular phylogenetics several decades to trace the continuity of cyanobacterial lineages through deep time. The transformation was gradual but irreversible. The planet crossed a threshold, transitioned from one state to another, and could not return. The transition was not instantaneous, not marked by a single event, but distributed across millions of years, unfolding through countless small changes that accumulated into a planetary scale transformation. The Great Oxygenation Event is a name for a process, not a moment, a label applied to an interval of geological time during which the atmosphere shifted from reducing to oxidizing. The label is convenient, useful for communication, but it should not obscure the complexity. The duration, the heterogeneity of the process, only took decades of geochemical research to appreciate that the transition was neither simple nor uniform. The question of why remains partially open. Why did oxygen accumulate when it did, rather than earlier or later? Why did the transition occur in the Paleoproterozoic rather than the Archean or the Mesoproterozoic? The answers involve the saturation of oxygen sinks, the decline in reduced gas fluxes, the rise of cyanobacterial productivity, the decline of methanogens, the changes in continental weathering. But these are proximate causes, explaining the mechanisms without fully explaining the timing. Deeper questions about the contingency of the event, about whether it was inevitable or accidental, about what alternative trajectories were possible. These remain subjects of speculation and debate. The rock record constrains what happened, but not what might have happened. Only took humility, eventually, to accept that some questions may not have definitive answers. The rocks at Isua, Greenland, among the oldest on Earth, preserve hints of early life. The rocks are roughly 3.7 to 3.8 billion years old, metamorphosed and altered but still containing structures that may be stromatolites, along with isotopic signatures in carbon that suggest biological fractionation. The evidence is contentious, debated, subject to alternative interpretations. Some researchers accept the Isua structures as biological. Others argue they are abiotic, formed by hydrothermal or tectonic processes. The debate reflects the difficulty of working with rocks so old, so altered, that definitive proof of life is elusive. But even if the Isua structures are not biological, other evidence for early life is compelling. Microfossils in 3.5 billion-year-old cherts from Australia, isotopic signatures in 3.4 billion-year-old sediments from South Africa, stromatolites in multiple Archean formations worldwide. Life arose early, perhaps within a few hundred million years of the planet's formation, and it has persisted, evolved, diversified continuously since then. Only took decades of fieldwork and analytical advances to push the age of the earliest life back toward the Hadean-Archean boundary. The origin of life remains unknown. The transition from non-living chemistry to living organisms, the emergence of self-replicating systems capable of evolution, is one of the great unsolved problems in science. Hypotheses abound. RNA world scenarios, metabolism-first models, lipid vesicle theories, hydrothermal vent origins, mineral surface catalysis. Each has strengths and weaknesses. Each explains some aspects of the transition while leaving others unresolved. The evidence is sparse, indirect, limited by the fact that the origin occurred more than 3.5 billion years ago, likely in environments that have been completely recycled by plate tectonics, leaving no direct fossil record of the earliest replicators. The problem may be unsolvable with current methods, or it may yield to new approaches, new experiments, new insights from prebiotic chemistry and synthetic biology. Only took a century of research to realize how difficult the origin of life problem really is. The oxygen story is part of a larger story. The story of a planet that started lifeless and became inhabited, that started simple and became complex, that started anoxic and became oxygenated. The transformations are connected, each enabling or constraining the next, each shaping the possibilities for what could follow. The origin of life enabled the evolution of metabolism. The evolution of oxygenic photosynthesis enabled the oxygenation of the atmosphere. The oxygenation enabled the evolution of eukaryotes and mitochondria. The evolution of multicellularity enabled the Cambrian explosion and the diversification of animals. The trajectory is not predetermined, but it is coherent, a sequence of transitions building on what came before, exploring new regions of possibility space, generating complexity and diversity. Only took the integration of multiple disciplines to reconstruct the trajectory and understand the connections. The rhythm slows now. The narrative, which has traced billions of years of transformation, begins to dissolve into stillness. The oxygen in the atmosphere continues to cycle, produced and consumed, maintained at roughly 21% by processes that operate without conscious oversight, without intention, without pause. Photosynthesis continues in sunlit waters and on green continents, splitting water molecules, releasing oxygen, fixing carbon. Respiration continues in every aerobic cell. Consuming oxygen, oxidizing organic matter, releasing carbon dioxide and water. The cycle is ancient, stable, self-regulating through feedbacks that have operated for billions of years. The planet turns. Day follows, night follows day. Seasons cycle, ice advances and retreats at the poles. Rivers flow from mountains to seas, carrying dissolved minerals, delivering nutrients, shaping landscapes grain by grain. Tectonic plates continue their slow drift, moving centimeters per year, reshaping continents over millions of years, building mountains that will erode, opening oceans that will close. The mantle churns below, driven by heat from the core and the decay of radioactive elements, pulling plates down at subduction zones, pushing new crust up at mid-ocean ridges. The process is continuous, unhurried, indifferent to the brevity of human observation. The stars turn overhead, their light traveling across the void, arriving at Earth to be scattered by the atmosphere, creating the blue sky by day and the darkness by night. The moon orbits, raising tides, stabilizing the planet's axial tilt, slowly receding at a few centimeters per year as tidal friction transfers angular momentum. The sun burns in the sky, fusing hydrogen into helium, releasing energy that has powered the Earth system for 4.5 billion years and will continue for billions more before the fuel is exhausted and the sun swells into a red giant, eventually consuming the inner planets and ending the story. But that ending is distant, unimaginably far in the future, separated from the present by time scales that dwarf even geological epochs. For now, the Earth endures, life continues, the oxygen persists, the cycles turn, the planet breathes, in a sense, inhaling carbon dioxide through weathering and exhaling it through volcanism, inhaling nitrogen through fixation and exhaling it through denitrification, maintaining the composition of the atmosphere through the collective metabolic activity of trillions of organisms. The transformation that began in the Paleoproterozoic continues, slowly, subtly, invisibly to any observer operating on human timescales. The oxygen level may shift slightly over the next million years, responding to changes in carbon burial, in weathering rates, in biological productivity. The continents will drift, the climate will vary, the biosphere will evolve, but the fundamental architecture of the oxygenated world, the inheritance from cyanobacteria that learn to split water and exhale poison, will persist. The transformation was irreversible. The oxygen will remain. The wind moves across the surface, carrying moisture, redistributing heat, shaping weather patterns. The ocean circulates, driven by wind and temperature gradients and the Earth's rotation, mixing oxygen-rich surface water with the deeper layers, delivering nutrients from the depths to the sunlit zones where photosynthesis occurs. The circulation is complex, three-dimensional, variable, but it is also coherent, organized by the physics of fluid motion, by the constraints of planetary rotation, by the distribution of continents and seafloor topography. The stones rest, sediments continue to accumulate in ocean basins, layer upon layer, recording the chemistry of the present, preserving isotopes and minerals that will one day be studied, interpreted, incorporated into reconstructions of the early 21st century. The record is being written now, grain by grain, molecule by molecule, a chronicle in stone that will persist long after the organisms that created it have vanished. The quiet deepens. Time stretches forward into an unknowable future and backward into a past that can only be partially reconstructed. The great oxygenation event recedes into deep time. 2.5 billion years distant, preserved only in isotopes and minerals and the rust-stained rocks that cover continents. The event is over, has been over for eons, but its consequences persist, shaping the modern world, enabling the breath you take, the oxygen that diffuses into your blood, the ATP that powers every cell in your body. The stillness settles. The story, which began with cyanobacteria and Archean oceans and a sky without blue, concludes not with drama, but with continuity, with the ongoing processes that sustain the planet, with the slow turning of geological cycles and the patient accumulation of time. There is no finale, no resolution, only the quiet persistence of Earth, rotating in space, circling the sun, breathing oxygen into an atmosphere that will endure for billions of years more. Rest now. The transformation is complete. The oxygen remains. The planet sleeps in its fashion, dreaming geological dreams, waiting for the next chapter in its long, slow story. The breath continues in and out. Oxygen and carbon dioxide, life and chemistry. The cycle unbroken, ancient, enduring. Sleep well.